Oh, hello, freaks. Yes, we are back again. Did you miss us? No, you couldn't have missed us. We've been doing this shit all week. This is ridiculous. This is the longest run of podcasts ever for me. There's 4 of them in the top 10 right now.
Yeah.
4 out of this. That's crazy. We have 4 podcasts. That sounds like bragging.
It's a lot of pressure for me.
This episode... No, there's no pressure. Dr. Patrick, worry not. This episode of the Joe Rogan Experience is brought to you by Stamps.com. Do you remember back in the days of black and white TVs and phones with wires? The only way a business could avoid the post office was to lease a postage meter. That's annoying. But today, technology has made it so that anyone can have that possibility at your home. All you need is Stamps.com. That's coffee. With Stamps.com, you can print official U.S. postage from your home or office computer right from your desk. Just bang. Just measure it out, print it out with a free digital scale that they provide, and you print out official U.S. postage. Mailman comes, takes it. You avoid all the lines of the post office. Official U.S. postage for any letter, any package, using your computer and printer. No extra hardware to buy or lease. And unlike a meter, there are no long-term commitments, and you can save up to 80% versus a meter. Plus, with Stamps.com, you can track packages, track spending, and more. We use Stamps.com. Tom Segura and Christina Pazitsky use Stamps.com. Brian Redband uses Stamps.com to send out the DeathSquad.tv t-shirts that he sells. And with this no-risk trial, using the promo code JRE, you will get a $110 bonus offer, which includes a digital scale and up to $55 of free postage. So do not wait. Go to Stamps.com, and before you do anything else, click on the old-school microphone in the upper right-hand corner and type in JRE. That's Stamps.com and enter JRE. Save yourself some money. And that's the only goddamn commercial we're doing today. How about that? Cue the music, young Jamie. Dr. Rhonda Patrick is here. We're going to find some shit out about health.
Joe Rogan Podcast, check it out.
The Joe Rogan Experience.
Train by day, Joe Rogan Podcast by night, all day.
Hello, Rhonda.
Hello, Joe.
You're the only person that's ever come in, sat down, and said, "Oh, this is good. I'm getting a cortisol rush." You said you're getting a cortisol rush from the feeling of about to do a podcast.
Yeah, actually, I could have gone a little more detail, but I was like,
Please do.
Yeah, so that feeling of anxiety, that like you get before you get on stage. I mean, you're a comedian. I'm sure you're really familiar with that. You actually are expressing a chemical called dynorphin. And this is an endogenous chemical in your brain, and it binds to something called the kappa opioid receptor. It's kind of like the counter of the endorphin, which binds to the mu opioid receptor. So the endorphins are the feel-good. Well, its nemesis is the dynorphin, which is that anxiety-like feeling. So, but here's the interesting part. That anxiety-like feeling that you feel before you're, going to do something like you're, really that you care about, or that's kind of anxiety provoking. For me, public speaking does it. I get it right before I'm going to do it, give a talk or something, or do a podcast in front of, 500,000 people. So that dynorphin binding to the kappa opioid receptor, what it does is it upregulates the mu opioid receptor so that after you get that anxiety feeling, you have a better endorphin rush because you upregulate the mu opioid receptors, which bind to the endorphins, the feel-good ones, and you actually become sensitized to them. So there's a biological mechanism by which having that anxiety actually does you good. So it's kind of like a stress at first, but then later on you feel better and you're more relaxed.
That's amazing. That's interesting because one of the best feelings ever is after you compete. Like when you fight, one of the things that people get addicted to is you get addicted to the rush, for sure, the adrenaline rush, and you get addicted to the challenge of competition being life magnified on such an incredible scale that everything else seems kind of pale in comparison. But the big addiction, I think, is to the feeling that you get after you compete, especially if you win. There's just an amazing feeling like life is incredible. You like the ground feels better under your feet. The colors and the trees look better. Everything looks better for a couple days.
Yeah, no, I totally know what you're talking about, and there's a biological mechanism for that. And it's interesting because it's also the same mechanism that occurs when you are working out really hard. I mean, it's painful while you're doing it, but the more pain you experience, the better the rush, the better. It's the endorphin high people talk about. It's actually that mechanism is anxiety-inducing, stress-inducing is an important factor in that because of this mechanism I talk about where you upregulate those receptors that bind to the endorphins and you're sensitizing them, so they become more sensitized to them. So it's heat does the same thing, stress. So, exercise capsaicin, so eating spicy food, it's painful while you're eating it. But what happens is you're upregulating those mu opioid receptors and you're sensitizing them to the endorphins, which is why you feel really good after eating spicy food.
Oh, that's what that is.
Yeah.
I never knew what that was. That's interesting because I always thought it just tasted good, like a kind of a weird, cool, good, spicy taste.
Oh, I'm sure there's dopamine responses and all that stuff involved. There's complex mechanisms and things going on in your brain, but when you're eating or when you enjoy a certain taste of a food. But this particular mechanism where you've got this dynorphin and endorphin, connection, it's really interesting, and I think it's something I discussed in a recent article that I wrote about the sauna. So I hope people find it interesting because I think it's interesting. It's not something people know about.
It is. It's gotten some really good responses too. I've read it and I've read a lot of the responses. People found it pretty fascinating when you were talking about it on the podcast. But it's kind of almost like the yin and yang of life is what you're talking about here. It's like you need to have some negative in order to feel some extra positive.
Right. No, are you familiar with hormesis?
I know, I've heard the term, but I couldn't describe it if somebody asked me to.
So what it means, you mean?
Yes.
Okay, so hormesis, what it really refers to is having a little bit of stress, something that's a little toxic for you, something that's a little stressful on your body. I mean, exercise for sure is that. Heat is a little stressful. Things in some of the foods we eat, like the EGCGs in green tea or polyphenols in some of the fruits and also in red wine. These things are actually a little bit toxic to our body. And what happens is that this induces stress response mechanisms in our body, the activation of a whole host of genes like antioxidant genes, glutathione peroxidase, things like heat shock factors, which do a variety of different things. So a little bit of stress activates this whole stress response mechanism in our body. And then what happens is now we can deal with stress better. So it's kind of like interesting to think about adding a little bit of something that's toxic to, get a better response.
Oh, that's fascinating. You were talking about that last time we were here about mycotoxins, that in fact mycotoxins in a small dose might induce that sort of a response.
Yeah, I was hand waving. I haven't read anything,
Get you allowed to do that.
I was hand waving. I'm like, well, actually, maybe it makes sense that in a small dose, I mean, obviously you don't want something that's like very toxic and going to kill your brain cells or anything like that. But things in small doses, and I was assuming that maybe mycotoxins could possibly have a little slight hormetic effect. Like I said, that was just me theorizing. But
isn't it fascinating though that is sort of the way, like especially when it comes to exercise, that you need to experience this like intense hardship in order to get this wonderful feeling of accomplishment, this endorphin rush, and this. I feel so good after I work out. Like I make all my best decisions after I have like a really good workout. It's just like my body has a perspective. I have a better perspective. If I haven't worked out in a couple of days and I'm stressed and then I have to make a decision, oftentimes I don't trust my judgment.
Oh yeah, I'm the same way. I'm absolutely addicted to the feeling and the physiological changes that occur with exercise. And there are a lot of them that are occurring, in the brain and elsewhere. But it's the same, what's really kind of fascinating recently, we know about inducing neurogenesis when you work out. That's kind of well known, where exercise induces the growth of new brain cells. BDNF is one way, IGF-1. These things can, you grow new brain cells. And so growing new brain cells has always been associated with learning and memory. It's like, oh, you grow new brain cells, and they've shown that neurogenesis, in adults at least, is associated with new learning and new memory. But what they found recently, and I found this really interesting because it kind of changed the way I think of it, is that when you grow those new brain cells, like when you're exercising, heat stress does the same thing actually, so getting in the sauna, when you're exercising, you're heat stressing your body. I mean, your core body temperature is elevated. It's, very similar. So what happens is you grow new brain cells. and these new brain cells have to make new connections with other neurons. And so what ends up happening is that other connections that you made with so the connections you make between neurons, these synapses that you form, memories. It's things that you can remember, right? I mean, you form these synapses, and that's like a piece of information you remembered, or in some cases, it's an experience that you had that you remember. Well, you actually change those synapses, and some of them go away, so you kind of forget to make room for a new neuron to make a new. So it really is interesting how also you have this connection, but it's a balance where you're forgetting things, like you're totally disconnecting some connections between old neurons that you had previously made for whatever thing you had learned, and you're making room for this new neuron that just was born to make a new connection and learn something new. So you have that balance. Again, it's just, it seems like a very important biological mechanism, this yin and yang, where it's like there's stress, and then there's the response to the stress, which is good, and there's things like forgetting things, but so that you can learn new things. I mean, it's kind of important. Can you imagine if you remembered everything you'd go insane.
Yeah, I have a real problem with that. I have a real problem with having too much information in my head and then forgetting things that other people remember, and they'll remember it like it was this big important thing, and I'm like, what? Did we really do that? I don't remember that. And it's just because I've had too many experiences. Like, there's too much stuff. Or if you go back to talk to someone you went to high school with, people that don't leave, that are stuck in that town, and they can remember when we were 17 and we did that crazy thing. You're like, no, I don't fucking remember that. Like, how do you remember that? Like, you got to get out of this town, man. You got to make some new connections in your brain, son.
Yeah, no, that's you're referring to what's called episodic memory, which is the type of memory that's associated with experiences. And there's a lot of different things that can regulate that. One would be like a very strong emotional response, like your amygdala is activated. So if something, you'll probably remember things that are fear-inducing or something that's really exciting, like really, really exciting to you. That those sorts of things can solidify those types of connections. So also serotonin plays a role in that, by the way.
Does it really?
Yeah. I've been doing this other paper that I've been working on. I've been looking at the role of serotonin in brain function and dysfunction and also behavior. But it seems like serotonin plays a role in executive function and specifically in episodic memory. So any episodic memory that has a big rush of serotonin, would it be because what would cause that specifically? Like, what would make a serotonin rush in a memory? Yeah, I mean, there's a lot of different things. Serotonin release happens to a variety of different factors, but one could actually be very potent, something that's very potent that you're experiencing. Does that make sense?
Yes.
Where it's like, it's not just your everyday stuff that you kind of block out, like it's just routine, but it's something just novel and
would that be like the day you find out that Kennedy got shot, or the day 9/11 happened, or the day, like, extreme events? What response would that be?
I think that would be the amygdala response. Yeah, where your amygdala gets activated. But I think for the serotonin, it's more of they've done a lot of studies where they've looked at kind of like depleting it and what happens when you when you can't make it. Your episodic memory is bad.
Well, that's the thing with people who do MDMA, with people who party too much and do ecstasy, is the crush afterwards is their serotonin being depleted, right? That's a big part of it.
What's the mechanism of action of MDMA? I always forget.
We could pull it up. Well, MDMA supposedly has some function with rushing serotonin into your system, and it gives you this gigantic. Let's see if it gives you induce euphoria, sense of intimacy with others, diminished anxiety, mild psychedelia. Psychedelia is a weird word. Doesn't seem like it should be real, but it is. Many studies, particularly in the fields of psychology and cognitive therapy, suggest they use it a lot lately. It's been used a lot for PTSD, which is interesting because I've only taken it once. I took it once a long time ago. I never took it again. I never had a desire to take it again, although I had a very positive experience with it because the downside with the next day was just brutal. Like, I couldn't read. I remember I was in a coffee shop and I was trying to read a magazine and I couldn't focus enough to read a paragraph.
You couldn't focus.
There was nothing going on. My brain was just my brain felt like a dry sponge. Like,
wow.
It was just
So some things that happen when you release a lot of serotonin, into your synapses is that your body, because it's too much, there's more than, it's supposed to have, you'll start to downregulate other receptors, and there's like a negative feedback. So what would happen then is the next day you're not going to be as responsive to that serotonin, and that's something that's a big problem with some SSRI drugs and things like that, where you're basically preventing the serotonin from being remetabolized, taken back up, and so it sits around in these synapses for a lot longer than it's supposed to, and they've shown that the consequence to that is you actually downregulate serotonin receptors, which is what, serotonin receptor responds to the serotonin that you're releasing. So you can't respond to it, then, what's the point? So there's a lot of these feedback mechanisms that occur, and I don't even know all of them. It's very complex.
Yeah, it's listed in Wiki under the long-term effects that it causes a downregulation of serotonin reuptake transporters in the brain. The rate at which the brain recovers from serotonergic changes is unclear. One study demonstrated lasting serotonergic changes, tonergic. In some animals exposed to MDMA, other studies have suggested the brain may recover from the damage. But a lot of people that do it, they take 5-HTP to try to juice up their serotonin. They take it while they're doing MDMA, and then they take it afterwards.
You have to be really careful when you're combining multiple drugs that are affecting, like serotonin, for example, or that pathway, because then you can end up getting something called, I think they call it serotonin syndrome, where it's like people that are taking SSRIs have to be really careful and not take a bunch of tryptophan, or because then you can induce like a toxicity that's pretty dangerous. And I forget what all the
Yes.
What all the actual effects of serotonin syndrome are, but they're not good.
Yeah, not good at all. That's an issue with people also that are taking 5-HTP.
Yeah.
If they're on a serotonin SSRI,
right,
and they take 5-HTP, you can get this serotonin syndrome.
Yeah. The other thing about serotonin, we always think about it as being a brain, neurochemical, right? A neurotransmitter in the brain. But actually, the majority of the serotonin in your body is not in your brain, it's made in your gut.
Really?
Yeah. So I actually just published a paper. It was published the last time I was on this podcast, actually, my paper. I didn't know it came out in press the day that I was on this podcast. But so there's two different genes that convert tryptophan into 5-HTP. Okay, that's the rate-limiting step where you convert tryptophan into 5-hydroxytryptophan, and that's called tryptophan hydroxylase. Okay, and there's one enzyme called tryptophan hydroxylase 1 that's localized or located outside of the blood-brain barrier, and it's predominantly found in the gut, but it's also found in the pineal gland, which is actually separated by the blood-brain barrier. It's close to the brain, but it's actually separated, and it's also found in some of your T cells and placenta tissue. But and then there's tryptophan hydroxylase 2, which is the brain form, and that's found in the dorsal raphe part, the mid part of your brain. And there's also some expression. So your gut has neurons in it as well, enteric neurons. That enzyme, that's the neuron one. There's some of that also in your gut, but mostly it's the other enzyme. So there's tryptophan hydroxylase 1, tryptophan hydroxylase 2. They both make serotonin. So serotonin made in your gut actually causes GI inflammation, and they've shown this in like mouse models because what happens is serotonin made in the gut activates T cells, and the T cells have a receptor to it on their cell surface, and when they respond to serotonin, they proliferate and grow more. And so if you have a bunch of serotonin in your gut, you can get GI inflammation, and I think this has also been documented with people that take too much 5-HTP, as well as people with colitis and such. They've done mouse models where they knock out that enzyme, tryptophan hydroxylase 1, that makes serotonin in the gut, and it completely ameliorates the inflammatory symptoms in those mouse models. So the other enzyme in your brain. So this is where my paper comes in. I'm sort of it's a theoretical paper, but I found a mechanism. So a theoretical paper means I didn't actually do any experiments on mice. I didn't actually do a clinical trial. I found an underlying mechanism, and I explained it all and found all this stuff that was buried in the literature and put it all together. It's actually my first theoretical paper that I've ever written and published.
Congratulations.
Yeah, thanks.
That's
so what I
awesome.
found was that these two enzymes for tryptophan hydroxylase both have what's called a vitamin D response element in it, which is a telltale sequence that vitamin D, when vitamin D binds to a vitamin D receptor, it recognizes that and it can turn on a gene or it turns off a gene. And when it turns on, the gene does what it's supposed to do. When it turns it off, it's almost like it's not there. What I found with it is that so the sequence itself of this vitamin D element can determine whether or not it's going to turn it on or off. And I found these two different enzymes of tryptophan hydroxylase had different response elements. One was an on signal and one was an off signal. The one in the brain was an on signal, and the one in the gut was an off signal, suggesting that vitamin D was regulating the production of serotonin in opposite directions in different tissues. So you needed it to make it in your brain. But if you had enough vitamin D, it would shut off the gut one, not completely, but turns it down so that you're not making as much serotonin, you don't have as much GI inflammation. And then I related this to autism because I know I'm getting way out there, but it's pretty cool. So autism has been on the rise. Like right now, the most recent CDC report that came out said that 1 in 68 children have autism. I mean, it's risen like 600% since the 1970s. Like it's astronomical, like how much it's risen. So they haven't identified a genetic cause. Like over 70% of autism cases have not been linked to a genetic mutation, which means to me, something in the environment seems to be going on. There's something that is, interacting with possible genetic mechanisms that's causing, this autism rise. And, vitamin D is one thing that's been, people getting adequate vitamin D has been on the decrease, the same time that autism's been rising. And so I came up with this theory that low vitamin D during pregnancy and also during neonate when you're a young child can lead to serotonin deficiency in the brain. And what happens is during fetal brain development, serotonin is critical to guide neurons to where they're supposed to go, to make them proliferate. It's an important differentiation factor to help them make the kind of neurons they're supposed to make. It plays a very, very important role in regulating brain structure and morphology.
What vitamin D in particular? Because vitamin D3 was the one that you were harping on last time.
Right, vitamin D3. So vitamin D3, the way it works is you can make vitamin D in your skin. D3, you can convert it into D3. Yeah, so UVB radiation. You need UVB light, and it converts something in your skin called 7-dehydrocholesterol into vitamin D3. And then this gets released into the bloodstream, goes to your liver, where then it's converted to 25-hydroxy vitamin D, which is what people, that's a major circulating form of vitamin D, and that's what you get measured when you go, get your vitamin D levels measured. And then it goes into your kidneys and gets activated to an active steroid hormone. And it's called 125-hydroxy vitamin D. So vitamin D gets converted into an active steroid hormone, and once it's converted into that active steroid hormone in your kidneys, what then it does is it binds to a vitamin D receptor in different tissues in your body, including your brain, and it does this thing where it turns on or turns off over 1,000 different genes in your body. So it's regulating a lot of different processes. One of them is the serotonin. So I basically came up with this theory that women were not getting enough vitamin D, and the child's brain was then becoming serotonin deficiency. It was changing the structure of the way the brain was developing, and they've seen now that autism actually seems to be occurring in utero. And they're finding now, they've been doing these studies where they're finding now there seems to be a good, strong environmental component, and they haven't figured out what it is. So I'm hoping that people will start. I'm not an autism researcher, and I'm not a neuroscientist. I mean, I know a little bit about it. But what I did was I kind of just took a step back and started putting all these things together and came up with this hypothesis. And I also found things like estrogen can activate that same gene in tryptophan hydroxylase. So estrogen can protect you. If you have low vitamin D, you can still make that serotonin from tryptophan because estrogen activates that same gene also. So, and then the whole gut inflammation thing, autistics have this, and I think that they have high gut inflammation because they have too much serotonin going on in the gut.
That's a common thing with.
It's common with autistics.
Wow, that's interesting. That's fascinating.
So it's pretty interesting. I totally forgot why I got on this. I totally
Well, we were talking about serotonin, 5-HTP, 5-HTP T cells in the gut.
Right. So that's another thing where it's like 5-HTP, the first thing it does is it hits the gut, and you've got that enzyme in there that can, in the gut, so 5-HTP actually bypasses the tryptophan hydroxylase part. It's now the next step, which is to be decarboxylated into serotonin. So you can actually convert it. If you take too much of it, you'll convert it into serotonin in your gut before it gets to your brain. So It's something to keep in mind. Serotonin does not cross the blood-brain barrier. 5-HTP does.
Wow.
So if you're converting the serotonin immediately in your gut, then it's not going to get to your brain. So there's complex mechanisms.
Does tryptophan cross the blood-brain barrier?
Tryptophan does get transported into the brain, yeah. And we talked about this a little bit last time, where it competes with branched chain amino acids to get transported into the brain. So what happens is, leucine, isoleucine, these things actually win. They both, because they're the same, they're going through the same transporter. Tryptophan is a very rare amino acid in proteins. It's much less abundant than branched chain amino acids like leucine and isoleucine.
Is it a myth, the tryptophan turkey thing, that correlation between eating turkey and getting tired after you?
I don't think that getting tired after eating it is a myth. I think it's a myth that it's due to tryptophan, because actually, so the pineal gland converts serotonin into melatonin, okay? And the pineal gland it's not part of it's separated by the blood-brain barrier. So that competition, for the branched chain amino acids getting into the pineal gland we're talking about something different. So, you probably are getting a little bit of tryptophan being converted into melatonin or serotonin and then melatonin when you're eating turkey. But turkey has a lot of branched chain amino acids as well. So
it's most likely just a fat fuck.
It's most likely, yeah, exactly. But you said it, not me.
Stuffing and all the other stuff that goes along with it. Another thing, the T cells, I want to get back to that real quick, because you were talking about the elevated T cell count.
Yeah.
Now, is that something that would help people that have HIV? Would a lot, like a high dose of 5-HTP, help people who have HIV that have low T cell counts? Does that make sense? Is it
That's
the same
interesting.
thing?
I've never thought about that.
That's an issue, right? With AIDS patients.
Yeah, there's a lot of issues. But yeah, they definitely do have low T cell. I don't know. That's a really good question, Joe. Actually, I'm pretty impressed. That's interesting. It's possible.
But it seems like 5-HTP, being converted to serotonin. Does serotonin have an effect on T cell counts?
Yeah,
only gut level serotonin.
No, it has an effect on peripheral blood T cell counts. Yeah, that's why it's actually a pretty fascinating question. I don't know the answer to, but certainly something that I think I'll probably look into after this.
Yeah, that seems like
podcast.
we have a supplement that we sell that is hold on, I'll grab it. It's in this box right here. That's 5-HTP, L-tryptophan, and a couple other things. It's called New Mood.
Yeah.
And it's basically. Oh, you got one right here. Yeah. That's a 5-HTP supplement that was originally created. It was originally, when we first came up with it. Allegedly, my friend and partner allegedly likes to do ecstasy, allegedly. And he came up with this, they called it roll off. And the idea would be when you came off of, when people do MDMA, they call it rolling. And when you're off the MDMA, you're like, oh. Well, that was the nutritional boost that you needed to get your brain to produce more 5-HTP or to produce more serotonin, rather.
Actually, what's really interesting as I look at this is that you have vitamin D3 in it.
Yes.
Which really surprises me, because whoever made this connection for putting the vitamin D in with the L-tryptophan, I'm pretty impressed, because that, until my paper was published in February, no one had known that vitamin D can regulate. I'm sure. I don't know. Whoever made that connection, that's interesting that you were able to figure out to put vitamin D with L-tryptophan, because I haven't seen any supplement on the market that's done that. I don't know if whoever did this knew, but because tryptophan, you need vitamin D to convert tryptophan into serotonin. So that's actually I'm interested.
Well, my partner Aubrey, who's also my good friend, is he's very diligent when it comes to the research involved in any of these supplements, and he's also been a freak about vitamins and nutrients like his entire life. His mother's a maniac when it comes to that stuff. He learned a lot of it from her, and then extensive research. But yeah, we made sure that when we were putting something like that out, we wanted to make sure that we had all the bases covered.
Yeah, no, that's kind of cool that the vitamin D's in there. I wouldn't have expected that.
The pineal gland is a fascinating gland. That the fact that is what creates so many different elements that regulate mood, and that also now, I don't know if you're familiar with this. There's always been this thing about dimethyltryptamine, which is the psychedelic drug that's produced by the brain. It's also produced by the liver and the lungs. Well, they've found recently, like within the last year, evidence that it's produced by the pineal gland. It's always been like this big. There was anecdotal evidence, but there was never real hard evidence until they actually showed that in live mice, the pineal gland is producing dimethyltryptamine. And that's the third eye. I mean, the pineal gland, in reptiles. So
it's producing it when you sleep, or?
Well, it's producing it during different periods of stress, during REM sleep. But it's hard to get hard to get a mouse to sleep while you got a fucking big saw mark on its brain, or an opening in its brain, and you're testing its pineal gland.
Right.
So the amount of evidence they have about humans, about when and where, is kind of limited.
Yeah.
But now they're making these correlating. They're making a correlation based on the mammal, based on the mice, but they're trying to do more studies on human beings. More accurate studies, and trying to come up with more accurate ways of testing, especially during different stages, because the hypothesis is the theory is that when during heavy REM sleep and during periods of extreme stress, when your body believes that it's going to die, like if you're under extreme physical trauma, like people that have had extreme physical trauma, and they have that go to the light moment, the idea is that that's a dimethyltryptamine rush. And that the reason why they have these intense afterlife experiences, and I came back and it was amazing. I got to see my mother, and I went to this place, and God was there. Well, you can get there if you take psychedelic drugs. I mean, you can get there right now. A healthy person can have almost the exact same type of experience with psychedelic drugs, and it's just a crazy psychedelic drug because your own brain makes it. So
the dimethyltryptamine that you produce in the pineal gland crosses over the blood-brain barrier and starts affecting things like in your brain. Is it binding to different receptors and stuff in your brain?
That would be a question for someone far smarter than me. But the drug, ayahuasca, I'm sure you've heard of that, right?
Yeah, I mean, I've heard of dimethyltryptamine as well, and I'm not well versed in the whole field.
Well, it's just a crazy drug because it's the most potent psychedelic drug known to man, and it's the one produced by your own brain. So it's really weird.
Is it produced is it like from melatonin, or is it completely different?
Completely different, but it's very similar to melatonin in its chemical structure. It's related, like they're close.
It's weird.
Yeah.
I mean, because melatonin, regulates the sleep-wake cycle and the circadian rhythm. So, I'm wondering if it's very similar in structure to melatonin. Would it have any role in that as well? With the circadian rhythm.
It could. I think they're all connected. I mean, they tried to this is when you look at any of the studies that they've done on the human mind, and the studies that they've done connecting various hormones and things like whether it's dimethyltryptamine or melatonin or tryptophan, I think it's probably pretty safe to say that we know a lot about that stuff, or they know a lot about that stuff, but they're not entirely certain about all the different effects that these regulating hormones have on the brain. Is that safe to say?
Oh, yeah, for sure. It's very complex.
I think they're probably the most ignorant about dimethyltryptamine. Because it's it's been thought of as a recreational drug for a long time.
So people aren't studying it as much, probably.
Well, the only guy that I know that's had grants to study it and has been allowed to study it by the Food and Drug Administration, or who regulates? I guess it would be the FDA that would regulate that.
Now, in terms of like the recreational drugs, I don't know.
I think it's the FDA because dimethyltryptamine is a really weird one because although it's illegal, it's in so many different things. It's impossible to get the sources illegal because dimethyltryptamine is in like 1,000 different plants. It's in grass.
Wow.
I mean, you could get grass. You could literally get phalaris grass. You just go mow a lawn, a phalaris grass lawn, take the bag of that shit, bring it to a lab somewhere, and you got DMT, and you have a Schedule 1 drug. I mean, it's just bananas.
My cat, well, she passed away recently, but she liked to go outside and chew her some grass. Like, I mean, it was like, and she would get real crazy about it too. It was like, like.
Well, you want to see something really crazy? You want to see a jaguar trip out? Pull that video of a jaguar tripping out on DMT. Jaguars in the Amazon find these DMT-rich plants, and they chew them and eat them and have psychedelic experiences. And they have apparently they have a different way of processing things in their stomachs than we do because they're primarily carnivores. Mm-hmm. So they don't have the same gut enzymes and things. Like here's the jaguar. We'll play this for you, then we'll discuss it. But it's so crazy because this jaguar, like, they do it actively. It's like something they do on a regular basis. And they've observed them many times. They'll eat these grasses and these plants, rather. And then they just trip their balls up.
Pet cats eat grass. Large cats, like jaguars, eat leaves. When regurgitated, they cleanse their digestive system. But, like catnip, some plants induce other effects.
This jaguar is, first of all,
Yagi is one of the commonest rainforest vines. It seems to cause playful, kittenish behavior.
Yeah.
But could something deeper be happening?
that cat is seeing all kinds of shit right now. They're the king of the jungle. I mean, the jaguar's in the real jungle.
Yeah, the brain's fascinating.
Yeah.
I mean, it's biology itself is pretty complicated. And
yeah, look at this jaguar. His eyes are dilated. This is the same exact plant that people use in ayahuasca rituals. So this jaguar's eating it, and he's lying on his back.
The jaguar also takes yachi to heighten its senses when it hunts.
It's pretty fascinating stuff.
Like so much of weird nature, there is still so much to know.
The human gut produces monoamine oxidase, which makes dimethyltryptamine. It kills it in the gut, so that if you consume grass that has DMT in it, you don't have a psychedelic trip. But I don't know if a cat has the same gut enzymes.
yeah, I'm not sure.
Yeah.
Yeah, no, your gut has a lot. I mean, it's making neurotransmitters. There's neurons down there doing stuff, and there's a connection between the brain and the gut as well.
But it's just the whole thing about the human mind is so fascinating, where someone like you, who is just sort of putting together all these different things that you found in different books, and you go, well, hey, look at this combination of things is going on, and the vitamin D and vitamin D depletion, and then here we have this issue with autism, and autism has to do with inflammation in the gut, and then has to do with vitamin.
I put together a lot of stuff, and I explained the male, dominance and all this. And really, my goal was, look, maybe I'm right on, maybe I'm wrong on some of these things, but I'm definitely right about some of these things. Mm-hmm. Here, I'm interjecting it into the world, the scientific world, for all of you people that actually do research on autism and do research on serotonin and all this, because I don't. I mean,
right.
That's not what I do. And I think that's. It's good to have theoretical papers like that sometimes, where it's like you get someone who can make all these connections, where there's tons of stuff buried in the literature. And I had a specific mechanism that explained it. And you kind of just say, here, now, if you think this is interesting, follow up on it. And actually, we started collaborating with a group that does do research on one of these, looks at serotonin and does vitamin D research. And they were just jumping out of their chairs. They were so excited. And so now they're doing experiments. And have already found positive data, which is really nice and reinforcing. But so that's, that was my goal, is to kind of just make these connections, big picture, find a mechanism. Finding a mechanism by which vitamin D regulated serotonin was amazing, and putting it out in the literature. I mean, but the whole vitamin D thing, vitamin D3, is important, and I think it's something that most people are. 70% of the population is not getting an adequate,
70%.
70, yeah. Actually, we have someone reach out to me on Twitter after the last podcast, an artist, and he was like, "Look, I really like what you're doing, and I've been wanting to make a cool infographic, just basically summarizing everything I ever talk about with vitamin D. It's like all in one graphic." And he did it for me. And if you want to pull it up, it's on foundmyfitness.com.
Oh, yeah.
/vitamin hyphen D. And it's a freaking awesome infographic where
What's this guy's name?
His name's Jason Wright.
Jason Wright?
Yeah.
Right, Jason Wright.
Yeah. So he made this infographic where it's like basically I talk about how vitamin D regulates 1,000 different genes in the body. Yeah, here it is. So it starts off here. It's got this guy with all these like chromosomes in him with telomeres because vitamin D is important for telomere length.
Ooh, I want to talk about that too.
Oh, totally. And then if you scroll down, it's like, look, he's showing, 70% of the U.S. population does not have adequate levels. I thought that was really
I like
cool.
how he made the graph too. You see 10 people. And
Yeah,
you see
exactly.
3 of them have it and
Right.
7 of them don't.
Right. And then it goes on, and this is like your tag cloud where it's like all the things that like go wrong when you don't have enough vitamin D.
That's incredible.
Isn't that awesome? So, and it's like learning impairments, reduced serotonin, increased cancer risk, over 1,000 different genes in the body. So then you scroll down more, and it shows a telomere because it actually DNA repair enzymes are regulated by vitamin D as well. So if you're not getting enough vitamin D, you're going to have, you can see that break in your DNA. You're going to get damage to your DNA. And then as you keep going, it shows you, all the factors that regulate your vitamin D, like how sunscreen blocks UVB, so you can't make it with sunscreen. Melanin, which is an adaptation to prevent UVB, rays from burning you, actually also blocks your ability to make vitamin D. And body fat regulates it. So all these different body fat regulates the bioavailability of it. So it has to be released into the bloodstream to get converted into active hormone. Age regulates it. 70-year-old makes 4 times less vitamin D from their skin than their former 20-year-old self. All these things. Living in the northern latitudes, another thing. People that live in, I think it says above 37 degrees north, with the exception of the summer, they cannot make any vitamin D from the sun.
Wow.
So the solution is vitamin D supplement. And this is just a really awesome way to put all this great information, show it with graphics. People like that, and it's easier for people to understand. And I go into, what your level should be and all that. But I was really stoked because I've been wanting to do this for a really long time, and I have no artistic capabilities.
Yeah.
So
no, that's so cool.
someone just reaching out to me and wanting to help was was really cool. So
where is it on your website if you were looking for it?
I think if you sign up for my newsletter, I think
that's
it's
how you get it.
Yeah, I think that's how you
your
get
website's getting crushed right now. It's really hard to even get to.
Is
It
it?
took me 5 chances to get to it.
Wow.
Yeah, it's not.
So yeah, that's the vitamin D.
That's very cool.
Yeah, totally.
The telomere length thing. The reason why I wanted to get to that is because I had emailed you about this supplement that I had read about called TA65. And I had read about it because my friend Bobby, he emailed me about it, and his dad was taking it. That's Bad Bobby for folks on the message board. BAADBOBBY. His dad was taking it, and his dad started experiencing vision improvement. And I was like, that's fucking crazy. And so, what is this TA65 stuff? So I go on to Google, and I Google TA65. And there's all these crazy claims, and a lot of people are selling it. And, there's a lawsuit against it because there was a guy who was working for the company, a former executive, who is suing them because he says that it might have caused him to get cancer. And I mean, the idea is what they're saying. This is the company that sells it. This is the correlation between cellular aging and telomere length is rooted in solid research. Telomeres become shorter every time a cell divides, and when they are lost, cells can no longer reproduce. The enzyme telomerase, how do you say it?
Telomerase.
Telomerase can lengthen telomeres, possibly slowing or reversing degenerative diseases. In one study, mice genetically engineered to lack functional telomerase to lack functional telomerase showed brain degeneration and shrunken testes, but those effects were reversed when the enzyme was reactivated. And such findings have sparked a lot of hype and encouraged a cottage industry of companies that assess a person's biological age on the basis of their telomere length. But TA Sciences has taken the buzz further, and they sell a pill called TA65, which it says can lengthen short telomeres.
Yes. All right.
No.
Telomeres,
Yeah,
yes. But let me
telomeres.
explain. This
Please
is,
do.
I mean, so complicated, but I think I can shed some light. I think
Okay.
I can. So this is all right. Telomeres, every cell in your body has 46 chromosomes, with the exception of your, gametes, which have 23. But these chromosomes have your DNA. Okay, they're wound up with these histone proteins, and that's where your DNA is. And at the end of these chromosomes are your telomeres. The problem is that these telomeres have a funny structure where their DNA it's a repeat of TTAGGG, and there's like this structural defect in the DNA of the telomere where there's a big overhang, and like one of your strands of DNA is longer than the other one, and so when the cell divides and it's got to copy its entire genome with all those 46 chromosomes to make a new cell with all that same DNA. There's a little gap there at the end because it's like, oh my God, I don't have anything here. How do I make the new DNA? And so what happens is the next cell then has a little bit shorter telomere because there was that little gap that didn't get filled. And this is happening successfully over multiple generations, year after year after year after year. Each time it gets a little shorter, And that becomes a problem because these telomeres are actually protecting your DNA from damage, oxidative damage from unwinding, and also from your chromosomes. When your cell divides, they protect your chromosomes from fusing together and like getting all these funny abnormal translocations, which lead to cancer and other problems. So you don't want that to happen. And the problem is that most of our cells, with the exception of our stem cells and our lymphocytes, don't express this enzyme called telomerase, which is able to actually rebuild the telomeres. It's actually able to rebuild the end where that telomere little gap where we can't make it. So it's really cool. It's like, well, here's this great enzyme that can rebuild it, but why is it only in our stem cells and why isn't it in all of our cells? The gene is there, but it's just not activated. It's silenced. And the answer for that is, well, if you express too much telomerase in all these other cells, what happens is you can make a cell immortal. And cancer cells have actually developed, they're so smart, cancer cells are like the smartest thing ever. They've developed this capability to reactivate that enzyme to become immortal. But the really ironic thing is that it's the critically short telomeres. When your telomeres get to a really, really, really short stage, they go into this crisis where it's like, oh my God, what do I do? What do I do? And they either die or they, go into this senescent state where they don't divide, or they somehow can reactivate this telomerase. And at that point, if they reactivate this telomerase, you can, if this is a damaged cell that already has mutations and stuff, you're like, boom, okay, we're gonna keep you going now. So it's kind of paradoxical that the short telomere length is what actually can reactivate the telomerase to make it become immortal.
And make cancer.
Yeah, well, if you already have mutation, if it's already like an abnormal cell because you've already gotten damage and stuff to it,
right.
Yes, exactly.
So,
you can take a precancerous cell and say, here, keep going, grow more.
Whoa.
So this is the telomerase is in stem cells because your stem cells aren't dividing that much. They're usually just sitting around waiting they have to come out and divide and make more of a certain cell type, and then they go back into just waiting around. So they're not constantly, dividing, meaning they're not constantly at risk for more damage. And I can explain that later. But anyway, so the telomerase and the mice. This is kind of cool. So you talked about these telomerase deficient mice. Well, mice actually, it's really weird. They express telomerase in all their cells. Their telomeres don't get shorter, and yet they only live 2 years on average. And so in order to try to understand the biological mechanisms by which telomere shortening, what those effects are, you have to genetically engineer them to not express that telomerase. And what happens is over multiple generations, it takes like three or when you get to generation 4, then you start to see progeria types of effects where now these mice, they're born, this is the 4th generation of not having this enzyme. It takes like that long to actually get the telomeres to a really short level where it starts to have progeria effects, and they start to prematurely age. Their tissues start to, break down, and they have all these sorts of aging effects. And so what they've shown is they can reactivate telomerase at that point after 4 generations, and it reverses those biological effects. So the tissues become younger, the stem cells, are active and replenishing populations like they're supposed to. So it's kind of cool. It's like here you have this pro-aging mouse, you reactivate the enzyme, and it literally reversed the aging in these mice.
Would that have a possible benefit to children that have progeria?
So it depends on what type of progeria. There's certain ones, like there's Werner syndrome, which if you pull up, if you just Google Werner syndrome and image
Werner?
it,
W-E-R?
Werner, Werner. That's a disease that's involves short telomeres. So you basically you can't rebuild your telomeres, and it's excessive telomere shortening. And what happens is, if you click on the one with the 2 pictures of the women, by the time they reach puberty, like 15. No, not that one. There was, if you go back, there's it's like Filipino woman there, I think.
Left-hand corner.
There we go. Left-hand corner. Perfect. Yes. So like she ages normally up until the age of about 15. And then after 15, she's 40 years old in that right-hand picture.
Wow.
40. I mean, she looks like she's like
80.
Right. Or 90. Yeah, she looks pretty old. So in that case, it'd be interesting to see if having telomerase could help rebuild the telomeres. But like I said, because of the potential danger of allowing a precancerous cell to become immortal, I think there's a lot of people are careful about that, and that's why it's not like on the market right now where you're just.
But it is. People are selling it.
Yeah.
This is the thing. This TA65 stuff, and this is the lawsuit. Is that this guy who worked for them is saying that he got cancer.
So the thing with reactivating telomerase is if you generally are healthy, you don't have a lot of, your C-reactive protein's low, you don't have a lot of inflammation, a lot of damaged cells, then a lot of precancer cells, your immune system's getting rid of them. Reactivating telomerase in a normal cell is not really a bad thing. It's not going to make it a cancer cell. Mm-hmm. It's going to make it immortal. It's going to help, it not have critically short telomeres. The problem is reactivating it in a cell that has a bunch of damage in it. And so to me, measuring, looking at how much damage is in your cell, like we can do that. I do that right now in people. I take their blood cells and I look at DNA damage.
Look at mine. Check them out.
Yeah. You have to come. You can't.
What do I have to do?
You'd have to come down to the Bay Area and sign a consent form.
That's it? Just like you sign a consent form when you do a podcast?
Yeah.
It's a different form?
Kind of like that. Just to be in part of my trial. So
like, what do you do?
So what I do is I take blood. Well, I get an MD to take it for me, and so they draw the blood. And then I take the blood and I centrifuge it through this gradient where I can then isolate just the peripheral blood mononuclear cells, which are like mostly B and T lymphocytes. That's in some monocytes. So I don't have any red blood cells or platelets or any of those neutrophils. I don't want those, or macrophages or things like that. So, and I take those peripheral blood mononuclear cells and I look at the amount of double-stranded breaks in the DNA. So I can actually measure that. I can quantify that. And not only do I look at those double-stranded breaks in the DNA, so the double-stranded breaks, I think we talked about this a little bit last time. So just normal living. Here's aging for you in a nutshell. Just normal living. Your metabolism, your mitochondria are generating oxygen radicals, which, get to your DNA and you get enough of these oxygen radicals, it causes a strand break, and then you get them in parallel, you have 2 strand breaks. UV radiation does the same thing, but let's just forget about all the outside stuff. Like, forget about carcinogens, UV, smoking, all that. Just normal, living does this. And this happens every single day, And so your body has to repair that damage. And I talked about those enzymes that are magnesium dependent, and how magnesium is important for DNA repair enzymes, and 45% of the population doesn't have enough of that. Well, I'm looking at people that are obese. And those people usually don't have a really good diet, and they're most often magnesium deficient. Well, I don't want to say deficient because it's not like a clinical acute deficiency. It's like they're inadequate. Okay, they have inadequate levels of magnesium. They're not taking in, enough magnesium, which is 400 or so milligrams a day. So I look at their DNA, their strand breaks, and then I have this, because I think that they're not getting enough magnesium. I'm not actually measuring their magnesium. Someone else that I work with can do that, but I haven't found it necessary to do that yet. Then what I'm doing is I'm measuring the capacity of their body has to repair a known amount of damage that I induce. So I look at their baseline damage, see how much damage is there, and then I induce damage with an irradiator, and then I measure over time the ability of their own enzymes to repair that damage. And what I'm finding is that if I look at someone who's lean or obese based on BMI, then I'm seeing there's differences. I'm seeing that people that are obese, like have a BMI of 30 or so, they have a lot more of this damage in their blood cells. And not only do they have a lot more of this damage, their capacity to repair this damage is impaired. So, and it makes sense to me. You're talking about people that are eating poor diets. They're eating very macronutrient rich diets. They're eating a lot of processed foods and junk food, and they're not getting their micronutrients. Their essential vitamins, zinc, magnesium, these are required for like 300 different, enzymes to work in the body. And so I'm measuring one of those, and that's DNA repair, which is important to prevent cancer. So anyways, what were we talking? We were talking
TA65.
That's right. So I went on this whole. So that's aging. That's part of aging in a nutshell, and there's other points of it we can get back to later in the conversation. But so TA65 is. I was actually really impressed because I read a couple of the studies. So TA65, astragalus root, some Chinese herb or something, or that's what it's from. I believe from what I read, and. I think I'm saying it right, the astragalus root. So it has the capacity to activate telomerase. And there's two different papers that were pretty good. One paper was a clinical paper where they gave people varying doses of TA65, and I don't remember the exact doses, but they did a dose response. And they looked at a couple of things. A, they looked at the activation of that enzyme telomerase, and B, they looked at telomere length. So they started the trial baseline, measured telomere length and telomerase activity, and then they gave people these TA65, various doses of it, and what they found is that in a dose-dependent manner, the TA65 increased the telomerase activity. And not only did it increase telomerase activity in the very high doses in a subset of people, it actually increased the length of their telomere over baseline. I've never seen this before. Usually when you're doing, a lot of other things that affect telomere length, vitamin D is one of them. Vitamin D, the way vitamin D affects telomere length is totally different than the way TA65 does, because vitamin D is preventing DNA damage, inflammation, things that accelerate telomere shortening. TA65 is literally rebuilding the end of your telomeres. So it's possible to actually start and end up with a longer telomere, as opposed to, other nutritional factors that regulate just delaying the attrition of it. Does that make sense?
Yes.
So that over baseline, they had like a 40%, I believe, from reading that paper a while ago, a 40% increase in telomere length.
That's incredible.
It was incredible. It's incredible. So then I went and read the other study, which was the mouse study. And actually the woman, the lead investigator on that, I'm very familiar with her work. I was really close to doing a postdoc in a telomere lab, because I've been very interested in telomeres for quite some time. So I had been familiar with her papers, like her publications. And I was like, when I saw that it was from her lab, I was like, I know I'm familiar with her work. She's pretty good, pretty thorough. And So her paper, what she did was she took that same mouse model that I was talking about, where they, over successive generations, they knock out that telomerase enzyme, and they, over 3 or 4 or 5 generations, they start to get these mice that have really short telomeres that are aging, their tissues are aging quicker, and they gave those mice, like 3rd or 4th generation mice, TA65. And what they found was that giving those mice the TA65 was able to rejuvenate their tissue. Their tissue started to look younger, very similar to telomerase reactivation, not as robust, which isn't surprising. I mean, you're talking about reactivating the entire enzyme versus something that's just able to activate it. But the mice didn't get cancer. They didn't get any types of cancer. So, but like I said, now if you were to take a mouse model, knock out their telomerase enzyme, inoculate them with cancer cells, so give them cancer cells, and then give them the TA65, they're probably going to do that experiment. I mean, that's the logical thing to do next, just to make sure to really prove that it's pretty safe. So bottom line is, I was impressed with TA65, actually surprisingly so, and personally, I think there's always that risk. It's like, well, if you have a lot of precancerous cells, there's no telling. I obviously, if it was a big issue, more people would be coming down with cancer.
I wonder how many people are taking this stuff. I've only heard of a couple of people that I know that are taking it. I've read some folks online on a message board that I go to that say they started taking it, but I don't know enough
about how many I don't know how many people are taking it either. That would be my one concern, though, is that having a bunch of precancerous cells, reactivating telomerase in those cells, then pushing them, giving them the fuel they need to then make more cancer cells. Well, that's a possibility.
The people that own the company, the whole thing is really, really odd. I don't know exactly what happened, because apparently there was like some sort of a physical altercation with the guy who's suing them, and they're suing him because they're saying that they lost $2 million in sales because he said that he got cancer. Which is, wow. And they're also saying that if he had cancer, he had it before he started taking TA65. How the fuck could you ever know that?
Right. Well, but that's the thing. That's the thing.
Right.
If you have cancer before taking it,
right.
I'm not saying this is going to happen. I'm saying theoretically, reactivating telomerase could push those cancer cells to full-blown precancerous cells to full-blown cancer, because now there's immortalizing them and letting them survive and propagate and proliferate.
Yeah. So here's the question, and here's the big one. Is there a diet, a thing that you can do? The word is always, you need to have cancer cells can't grow in an alkaline environment. But I've heard that that's horseshit too. That this idea of alkalizing your blood and having a diet that makes your blood or your body alkaline rich is horseshit.
I'm totally not familiar with any of that. So
never heard of that before?
I've heard of people wanting to alkaline their blood. I didn't know why. I never really followed up on it, and I certainly haven't heard about the cancer connection to it. So that's new to me. I've definitely heard of other things people are trying to do.
Well, you can go alkaline cancer, and one of the first things that pops up is myth. The acid alkaline myth. If what I've read that's counteracting this is that if you did alter the alkaline of your body, like the variables are so small that if you alter it in any sort of a shocking way, your body just is fucked. Like it's not good.
Right. No, it's true. I mean, I think with the exception of the gut, where you're trying to actually make it a little more, you want these different gut bacteria to make more acidic, to be more acidic and make more, acidic type of environment to get rid of the bad bacteria, which can't grow in that type of environment. But all the other stuff, I think, there's slight changes and pH, and I mean, you're talking about activating these things. The immune system's sensitive to this sort of stuff. I mean, it's like all of a sudden you start activating your neutrophils, and they don't know why they're activated, but when they're activated, it's just like fire, 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 and they're firing all kinds of crap cytokines, which are making reactive nitrogen species, which are damaging your DNA and your lipids and your proteins. So it's like there's things like that going on. Anyways.
Well, this critique of it was saying that foods don't influence the blood pH. And they were saying, I'll just read what it says here on this guy's website. Proponents of the alkaline diet have put forth a few different theories about how acidic diet harms our health. The more ridiculous claim is that we can change the pH of our blood by changing the foods we eat, and that the acidic blood causes disease while alkaline blood prevents it. This is not true. The body this is all reference to, I'll give the guy's website. It's Chris Kresser, KRESSER.com. This is not true. The body tightly regulates the pH of our blood and extracellular fluid, and we cannot influence our blood pH by changing our diet with references. High doses of sodium bicarbonate can temporarily increase blood pH, but not without causing uncomfortable GI symptoms, and there are certainly circumstances in which blood is more acidic than it should be, and this does have serious health consequences. However, this state of acidosis is caused by pathological conditions such as chronic renal insufficiency, not by whether or not you choose to eat a salad or a burger. In other words, regardless of what you eat or what your urine pH is, you can be pretty confident that your blood pH is hovering around a comfortable 7.4.
Yeah.
So all that, you need to make your body alkaline, and cancer can't exist in an alkaline environment. Horseshit, right?
I just don't know enough about that stuff, honestly, to say if it's, I really don't. I know that things in vitro, when you're looking at cancer, if you're talking about growing cancer cells in a dish and changing that, environment is one thing, but I don't know. Yeah.
Yeah, I don't know either. That's the problem. God damn it.
Well, I mean, it's something. If you want to email me, I'll look into it.
Yeah, I will.
It's a matter of me not having dug into something like that.
Yeah, but that's it's one of those things that just gets repeated over and over again.
Right.
And by the way, this myth busting there's a bunch of different websites that say that it's horseshit. So, and a bunch of different doctors with references about this whole alkaline thing. Here's a cancer myth number 1, that cancer doesn't feed on sugar. Almost every new patient says, well, ask me about this. A theory is prevalent on the internet that sugar will influence blood sugar levels, feed cancer, and cause it to spread. The truth is you can't really control blood sugar by what you eat. The body's complex system processes what we eat and manages to keep blood sugar levels stable. Well, is that true? That doesn't make sense.
Well, I mean, when you're eating sugar, you're inducing an insulin response, which then you take the sugar up into your cells, so that regulates the blood sugar levels. Unless you're type 2 diabetic or something that's not working, then you can't regulate your blood sugar levels normally, and that's not good.
It's saying the exception to this, of course, is people with diabetes who don't have the proper insulin-regulating systems. But if you apply the theory that sugar can affect your insulin levels and feed cancer, diabetics with cancer would all be dying of their cancer. What?
What?
Yeah, exactly. What kind of fucking quote is that, Dr. Holen? Who are you? You fucking weirdo. That doesn't even make any sense. He's an oncologist, too. Specializes in gastrointestinal and esophageal cancers. Says at worst, these myths may lead vulnerable cancer patients to try untested treatments or procedures.
The thing is that cancer cells do acquire this capability to become glycolytic, where they're, instead of using glucose to convert it into pyruvate and go into the mitochondria and use this whole mitochondrial metabolism, oxidative phosphorylation, to generate energy, they use this whole pathway that's called glycolysis, which is it's a much shorter pathway, and they're using it to generate, that's how they're using it to generate their energy. So I think that probably plays a role in people coming up with all these different theories and things that manipulating the glucose and all that, what it can do. I know that, I did a lot of work manipulating cancer nutrients in cancer cells in grad school, and this is in vitro. First of all, if I do it in vitro and if I take a cancer cell and I take away their glucose, but they still have all these glutamine, they have these other amino acids, and they were fine. They'd grow slower, but they wouldn't die. If I took away their glutamine, they would die within like 24 hours. And it's, for me to think about cancer cells, well, not only do they need energy, ATP, they need nitrogen source to make new nucleotides for new DNA and also new amino acids. They need, molecules like to build lipids for your lipid membrane. So there's a lot of, just taking away the glucose is one thing, but there's a lot of other, macromolecules that are really required for cancer proliferation that are also important. So, taking some of the stuff that we're learning in science and immediately applying it, I think in some cases can be a little dangerous, just because we don't exactly understand how all these mechanisms are working together. So I'm a little, cautious about taking something that we're learning in science and immediately applying it to yourself, especially if you have cancer.
It's so fascinating.
If you think about, like, the last time I was here, I was talking about the folic acid.
Yeah.
And how that's a perfect, example where, folic acid is great for you if you don't have cancer because you can build new DNA. But if you have cancer, taking a lot of it is not great for you because you build new DNA, and that's what cancer cells are doing when they're dividing. So, and there's another example of that with another micronutrient, one of the vitamin E's. So actually, this would be a transition into some of the Offit stuff because he was talking about that specifically.
The doctor, that stuff that I sent you.
Yeah.
This is so we definitely get into that, and I would love to in a moment. So just to let everybody know, it seems, if you're not interested in doing any of the research, it seems that almost everyone who's done any research on this alkaline diet thing says it's horseshit. But they do say that these foods that they're suggesting that you eat to keep an alkaline-rich diet are very healthy for you. So in that sense, it's good for you because it's going to provide your body with the nutrients that it needs, especially if you're nutrient deficient. You're much more likely to be unhealthy. If you're much more likely to be unhealthy, your immune system is going to have a harder time dealing with any host of different diseases. But that this alkaline thing of your body basically hovers between 7.35 and 7.45. Your stomach is very acidic with a pH of 3.5 or below, so it can break down food. And your urine changes depending on what you eat. And that's how your body keeps the level in your body steady. But that this idea that you're going to regulate the alkaline of your body with food and that you're going to keep your body in an alkaline state, horseshit.
Well, there's a lot of those sort of like myths going on. In fact, recently I think I saw on your Twitter, you posted something, I don't know, a couple days ago about kale. And
yeah,
it's really kind of these people are doing more harm in a sense in some cases. So kale in the cruciferous family, like broccoli, cabbage, Brussels sprouts, these things, they have something in them called glucosinolates. Okay, and glucosinolates get cleaved by an enzyme called myrosinase, which is in the plant. And it forms something called isothiocyanates. And isothiocyanates are they've been shown to be very potent anti-cancer agents. So they activate, it's actually a part of that hormesis I was talking about because these are generated in the plant as a natural defense mechanism. It's one of their natural defense mechanism against bugs. It's like a natural pesticide in a sense. They produce it to keep bugs away. Isothiocyanates are, for us, activate a variety of different genes that are involved in stress resistance, including tumor suppressor genes, which actually kill tumor cells. And they've shown this. They've shown this, quite a few times that if you give mice, like mice that have cancer, isothiocyanates, they will kill the cancer cells. And the whole isothiocyanates thing also competitively binds to the iodine transporter in the thyroid. So here comes this whole, oh my God, you can't eat kale or cruciferous because it will screw up your thyroid and cause hypothyroidism, right? So the thing is, though, is these isothiocyanates, they appear to competitively bind to the same transporter that iodine does to get into the thyroid. Now, I was looking for the paper where they showed that, and it's like 1948, because what I wanted to see was a dose-dependent manner of giving them, these isothiocyanates or kale, and show how much of it, competes it with iodine and how much, doesn't. Well, I couldn't get access to that paper. But what I did find is that when you give these mice isothiocyanates, a lot of them, that kills cancer cells, but it doesn't cause any thyroid problems, which leads me to believe is that the isothiocyanates probably very, very, very small degree of competitive inhibition of getting iodine into your thyroid, which if you aren't low in iodine wouldn't be a problem because so if it's just very small amount of it competing with it to get iodine into the thyroid, it's not going to be a problem unless you're really, really deficient in iodine, and then any small amount you need. So in my opinion, and for me, I love getting isothiocyanates from these cruciferous. And by the way, the enzyme myrosinase that converts the glucosinolates into isothiocyanates is heat sensitive. So if you heat it, it will generally inactivate it, not completely, but it does inactivate it for the most part. And so you're not getting as many of those isothiocyanates, which is probably why people started to say, oh, boil your kale. Or I personally want the isothiocyanates because the data is so strong showing that these things kill cancer cells. I mean, it's a well-known fact that these things...
So this idea that raw kale or any of these raw what was the other?
There's another one.
Sprouts?
Okay. Brussels sprouts, but then there's another... So that's that story.
But the idea that those are bad for you seems bullshit.
Well, yeah, that's what I'm just... I mean, to me, it hormesis, okay, it depends on what you're defining as bad for you. Or saying hormesis, it's activating a bunch of genes that are tumor suppressor genes that kill cancer cells. I mean, it's good for you. These isothiocyanates are good for you if you're not like completely deficient in iodine, which most people aren't. I mean, I think that there's some problem with women in their 20s or 40s.
But if you supplemented iodine while you were taking these
right, and
isothiocyanates.
Isothiocyanates. They're very... I've been talking about these for years, like how awesome they are. I want them. Like I want them in my body.
Want them.
I want my isothiocyanates. So
you almost swore.
I'm telling you.
You got close.
You
said it.
So, because they're fucking awesome. It kind of irks me because I'm like, I see all this stuff and it's like, no, don't eat your raw kale because you want to get things like isothiocyanate. There's chemicals in these plants that are, yes, they're slightly toxic, but they're inducing stress response mechanisms in our body that are good for us. They're fighting off all the bad stuff that we're making in our body every day. It's a good thing. Precancerous cells are always happening. You want tumor suppressor genes are genes that when there's something's wrong, like you get that DNA damage I was talking about, something happens or a mutation happens in a cell, it senses it, and it activates this whole response pathway where it's like die and it kills the cell. So having something that activates those pathways in your body, like isothiocyanates, is a good thing. It's, and personally, I want more of them.
So all this stuff about this poorly researched, bad conclusions.
So the thing is when you look in the literature, if you're trying to look at the effects of isothiocyanates on thyroid function, there's not a whole huge literature on it. Like I said, I was looking for the experiment to prove to me. I wanted to see how much competitive inhibition there was of, I think they're also called IC3s on the iodine transport in the thyroid. I couldn't find it. I saw review articles where they referred to it. I looked at the reference, go to the reference. It's some other review. It's just, I wanted to see the data and I had to dig until I got to 1948 where they did this competitive binding assay. And then I found, other papers talking about, it's only an effect if you have low iodine, which made me think, well, I can't see the data, but what I think makes sense is that only if it's such a low competitive binding that it's not like if you eat this, you're going to not get iodine. It's that if you already are at that point where you don't have it, any little bit you don't get makes a difference. And I know this because the mouse models where they're not even looking at thyroid function, they're looking at the effects of isothiocyanates on killing cancer cells. Those mice have no problems with becoming hypothyroid, and they're giving them large doses. So to me, I'm like, okay, well, that would have been a side effect they would have noticed, So
this is just completely theoretical, and it doesn't work. It doesn't work in practice. This idea of getting hypothyroidism from...
I think if you have normal levels of iodine, you're fine. But if you're like one of those people that is iodine deficient, it may affect you, because if there's a small amount of it does seem to compete with iodine transport, but I think it's a very small competitive amount. It's not like all the iodine. It's not like all or nothing, like all of it's getting in or not getting in. I think it's a very small amount that doesn't get in. But if you're getting your iodine and it doesn't, are you following?
Totally.
Yeah.
Totally.
Okay. So that I hope people can follow that, because I really.
There's so much of that online these days. There's so much contrarianism. There's so many people that want to debunk anything that comes out.
That's the thing. And the other one is this oxalic acid. I think you kind of confuse the two.
Well, that was the one that Dave Asprey had talked about. That's why the oxalic acid, with kale. That's why I brought that up.
With kale?
Yeah, he had said that you had to boil kale because of oxalic acid and drain the water.
Okay, so let me tell you about that. The oxalic acid, I think there's a little bit in kale, but it's mostly in spinach. Oxalic acid's in spinach.
Much stronger in spinach.
Yeah.
Right? That's the highest quantity of it.
And what people are saying on the internet, from what I could see on this whole, echo chamber thing that happens, is that you need to boil your spinach, I guess, or in some people they're saying kale, because then you're going to inactivate this oxalic acid, and now it's not going to bind up minerals like magnesium and calcium. And they're saying that this is a problem if you don't do this, because then you're going to get kidney stones.
Right.
So I was looking in the literature, and I found a really cool experiment done by this Japanese group where they took spinach, raw spinach, they boiled it, they fried it, they frizzled it. So they did, various different temperatures. And they, then you make it into a powder to give to these mice that are magnesium deficient. They put them on a magnesium deficient diet, and they fed them either the raw spinach, the boiled, the frizzled, the fried. And they measured magnesium levels in their blood, in their bone, and they measured calcium levels in their blood and bone, and also in their kidneys. And what they found was that there was no difference in the magnesium and calcium levels in the bone and also in the serum, whether or not you boiled, raw, fried, whatever, frizzled. The calcium levels, there was a difference from frying it or frizzling it, but the raw and the boiling were the same. And basically, the conclusions were it doesn't affect absorption. Boiling or raw, it's the same. You're going to absorb the same amount of magnesium, calcium. There were magnesium and calcium in the kidney. They did have them in the kidney, and it was a little bit higher concentrated versus the control. But it wasn't causing, kidney stones and things like that. And there was no difference between boiling or not. So if you're going to make the claim that, having oxalic acid causes, kidney stones and you need to boil it, well, maybe you need to say don't eat it. But I don't think that's the case. I don't think any of that's the case.
So having oxalic acid in your diet, from consuming it in raw form, not bad.
I think you'd have to consume a massive amount to cause something that would be like kidney stone.
So what do you mean, like eating it every day, all day?
No, I mean like eating like pounds of it a day. Yeah.
Every day.
Yeah, every day.
Okay.
So
that's beautiful.
I'm not here's the bottom line. I have not convinced myself that I have to boil any of my spinach or kale before I eat it raw. I actually put it in a smoothie every morning. I make a smoothie with kale, raw spinach, tomato, carrot, avocado, banana, almond milk, some berries, and protein powder as well. Anyways, I haven't convinced myself that eating raw spinach in my smoothie every day, and I put a nice amount of it. It's not pounds of it, but I haven't convinced myself that I'm doing any harm.
And this is obviously not just based on the way your body reacts, but based on the research that you've done.
This is based on the little bit of reading that I've done on those. The isothiocyanates, I'm definitely convinced that, the oxalic acid, because it does bind minerals, it's a chelator. There may be some cases where kidney, can accumulate in the kidney. But I think that from my reading, that's a really, really large dose. And I haven't convinced myself yet. I'm not saying it's not possible, but I haven't convinced myself that I need to worry about it.
So the idea that you're getting some sort of toxins from the vegetables because the vegetables are trying to prevent predation.
Right.
Nonsense.
No, you are getting
but nonsense that it's bad for you.
They're activating hormesis, so it is kind of toxic for you. It's activating stress response pathways that are ultimately, it's like, okay, a little bit of bad for you, activating a pathway, really, really good for you, because now you're turning on hundreds of genes that are involved in DNA repair, that are involved in glutathione peroxidase, getting rid of oxidation, that are involved in making sure cancer cells die. So to me, you are getting a little bit of these toxins from these plants, absolutely.
But your body's counteracting of these toxins is incredibly beneficial.
Your body is your body is pretty amazing, and it is pretty beneficial, the reaction to some of these toxins.
So the point being that the incorrect correlation that people have made between the very low toxicity between these plants, kale, broccoli, whatever, and them being something that you should avoid consuming unless you boil them. That's just not doesn't seem to make sense.
In my opinion, it doesn't make sense.
Doesn't make sense. It doesn't make sense to me either.
So I think people can take something little and it becomes sensational, and they're like, oh, it's very sensational because what's good for you is bad for you. But if you do this little thing, it becomes good for you again. And I think that it's a good marketing tactic in some respects. Do you know what I mean? Where
there is one case, there's a study that you could find on a PubMed site about oxalate nephropathy due to juicing. And this patient with oxalate-induced acute renal failure that was attributable to a consumption of oxalate-rich fruit and vegetable juices obtained from juicing. We describe the case and also review the clinical presentation of 65 patients seen at the Mayo Clinic in Rochester, Minnesota from 1985 through 2010 with renal failure and biopsy-proven renal calcium oxalate crystals. The cause of renal oxalosis was identified for all patients, a single cause for 36 patients, and at least 2 causes for 29 patients. 3 patients, including our index patient, had presumed diet-induced oxalate how do you say that word? Nephropathy?
Yeah.
In the context of chronic kidney disease, identification of calcium oxalate crystals in a kidney biopsy should prompt an evaluation for causes of renal oxalosis. Blah, 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 blah. So what does that mean?
Yeah, it sounds like these people. How many of them were that had the from the juicing?
Let's see, 65 patients from 1985 to 2010. That's a long time, and to have 65 patients,
and all of them were from juicing.
Actually, single cause for 36 patients, and at least 2 causes for 29 patients.
So 2 causes, then.
Yeah, the single cause for the 36 patients they believe was juicing.
So there's an N of 1 that they knew it was juicing. Okay.
And the other one, there's 2 causes.
And the other one, there's 2 causes. So about 3.
Yeah.
So to me, it sounds like I said, there's always going to be cases where you can probably cause that to happen because it does bind and chelate these ions.
Especially in juicing, perhaps, because you're taking such a large quantity of vegetables and breaking them down to a juice. Would that make sense?
So.
You would get more oxalate?
Maybe
oxalic acid?
Maybe so.
Because, if you juice, think about, what a 16-ounce glass of juice, how many vegetables have to go into that juicer to liquefy it down to... I make smoothies for the most part. I don't juice. Occasionally, I buy cold pressed juices, but most of what I do, I just blend
You're like
them.
me. I don't juice either. I have not ever juiced. I make smoothies as well, but it sounds to me like there can be cases where you're getting a really concentrated amount of this, and you're doing it every day because there's people out there that go extreme, and no matter what they do, they go to the extreme, and because you can. The oxalic acid or the anion of it, the oxalate, does bind to these metal ions. It chelates them, and it absolutely could accumulate in your kidney. So
the real key is the thing that we've always been taught, to eat a balanced diet.
I think most people are not going to that extreme.
Right.
And I don't think most people have to be under a fear that they can't eat raw spinach. That's the thing. I don't think you need to fear eating raw spinach. I think maybe just don't be a dumbass and go way, way, way overboard.
Right. So, eating some raw juices is probably really good for you. Eating nothing but raw juices all day, all your life,
Right.
not so good for you. You
Right.
need animal protein or some
You
other
need some
form of
other
protein.
things, and yeah.
There was another guy. I know of a guy who Brock Lesnar was actually the UFC heavyweight champion who ate nothing but meat. He was a fucking crazy person, and he got diverticulitis, and he had to get 12 inches of his colon removed. Pretty serious operation.
It's really interesting that I don't know how much of a degree of this is dose dependent, but there's bacteria in your gut called putrefaction bacteria, and I call that because they're the bacteria that make that really nasty smelling fart. It's like hydrogen sulfide. It smells like pure hydrogen sulfide. And those bacteria, the putrefaction bacteria, they actually use like sulfate and nitrate as their source of energy. And they convert it into, like, for example, hydrogen sulfide. But they need, it's basically what we do with oxygen. They do that with these sulfate. And they make it into hydrogen sulfide. It's like us converting oxygen to water. That's how we make ATP. They do this by converting sulfate into hydrogen sulfide, and that's their energy, this putrefaction bacteria. Well, they need these, cofactors to do this, and that's heme. Heme is in red meat. And so some people that eat really, really large doses of red meat, like what happens is you're giving those bacteria heme, and they start to make hydrogen sulfide. And the thing with hydrogen sulfide, so first of all, you get putrefaction of bacteria. So you'll have nasty smelling farts, like hydrogen sulfide farts. And also hydrogen sulfide competitively binds to enzymes in our gut cells that make energy. So hydrogen sulfide goes and binds to these electron transport chain enzymes that make ATP in our gut cells and competitively inhibits them from actually being able to reduce oxygen to water to make ATP. So then your gut cells start to starve down there and starts to break down the gut mucus barrier. Anyways, it's totally a tangent, and I don't know how I got on that. I guess the meat, the high meat diet.
Right.
So
the diverticulitis that people got from eating only meat.
Yeah. So like I said, I think it's a dose, really dose dependent thing, because eating a little bit of meat, it depends. I mean, you know your own body. If you're eating tons of meat and you start smelling these hydrogen sulfide farts, and
that's not good, right?
Probably not.
Not good. So if you smell a guy who's farting like that, you should talk to him.
But it's not the kind of fart that it's the pure.
Yeah. Those ones that are like, oh, good God.
Yeah, it's that one.
Yeah,
stuff's going on. Yeah,
those are bodybuilding farts. If you're around any
makes
guys
sense
at the
because
gym,
they eat a lot of red meat, right? Do they?
A lot of protein. Period. Does it affect all other types of protein?
No, it's just heme. Heme is it's what gives red meat the red color.
Ah, I see.
Yeah, it's specific to red meat.
So the more red meat, the more horrible farts.
Pretty much.
That makes sense. Who do you think about having the worst farts? It's always guys with big guts. If someone farts on an airplane, you always look at the guy with the gut. It's very difficult to figure out who's farting on an airplane. It's one of the reasons why people are so sneaky about it.
Yeah.
Sneaky airplane farters. You know who you are. But the guys with the big guts, they're usually the culprit.
It's the putrefaction bacteria. Putrefaction bacteria. I mean, they're named for a reason.
Yeah.
And they're pretty nasty.
It makes you want to throw up.
It does.
So off it. This is, my friend Brian Callen, who I love dearly, but he's fucking ridiculous. And he's one of those guys that will get on this tangent and listen to one person say something and then spout it out as if it's fucking gospel.
Right.
And he hit me with this Peter Offit guy. Is that the guy's name?
Paul, maybe?
Paul, whatever the fuck. Dr. Offit, whatever the fuck his name is. And he listened to this guy, and without debunking it online at all, and I did it real quick. I did a real quick debunking of it and found a bunch of people who called bullshit on all of his claims. Meanwhile, these studies were from 1942, and it's just like so much of what this guy had said was horseshit. One of them, Offit, claimed that a study... What this guy was basically saying was that taking vitamins and taking antioxidants can actually cause cancer. And, Callan, of course, never wants to be wrong, so he's telling me, no, all studies confirm this. I'm like, what the fuck they do, man? So I sent him all this, I barraged him with all these debunking sites, and he backed off, finally. But for fucking days, his Twitter was all about don't take antioxidants. I'm really.
Oh, so you convinced him?
No, he fucking called me up. He's like, dude, I'm really worried about you. You're taking antioxidants, you're taking vitamins, and where are you going to get cancer? I'm like, the fuck are you talking about?
Oh my God. We need to dive into this. I mean, so
Please.
First of all,
it angers me.
What was the name of the book he wrote? It's Do You Believe in
It's
Magic?
called I'm a Cunt. That's his book. I'm a cunt and I'm trying to make money. Because I think that's what's going on with a lot of these fuckheads. These contrarians, with these people that make these articles. I don't know how many of them are just idiots. I don't know how many of them are like Brian Dunning, where they just sort of, they're mentally deficient. There's something wrong with the way their thinking works.
Yes.
And,
yes.
and I discovered that in communicating with Brian over 3 hours. I'm like, oh, there's something wrong here. And I don't know what it is. And I don't want to know what it is, but it's obvious. You're not normal. You're not healthy. Your correlations are not healthy. Just in discussing buildings falling, his way of looking at things is unhealthy. It's not correct. I don't know if that's what this guy's doing. I don't know if a lot of these people are doing things. I think they're just trying to get attention. They're like, oh, kale's gonna kill you. Fucking kale's gonna kill me. Everybody knows that people are loving kale these days. People are finding these health benefits. Kales. I talk freely and openly about how I drink kale salads or kale shakes rather on a regular basis, and how I feel great when I take them. So, and then, people will send me these fucking things. So I retweet some of them, and then I'll find contradictory articles. I retweet those. So this off-it fucking thing just drove me up a wall, because I started reading all the people that call bullshit on this guy, and all these people that say that he's kind of dangerous, that the things he's saying are kind of dangerous, because they're easily refutable, but he's trying to sell them. He's selling them as fact.
Yeah, so... let's dig in here, because I really first of all, I wasn't familiar with the guy until you brought him to my attention. And actually my advisor, my postdoctoral advisor, he's an 85-year-old like world-renowned scientist. He knew who the guy was, and he was like, you're going to crush him. Like he really doesn't like this guy. I mean, he's familiar with his whole, everything he's doing.
Well, that's why I was so angry at Callan, because if you go online, there's just a fucking slew of reputable people that think this guy's a nut.
So I didn't read his book, but I listened to the podcast and I took notes on what he said. And I'm ready. Let's go through some of this, because I think it's really important. First of all, this guy makes huge overgeneralizations, doesn't understand mechanism. That's clear to me. So the first thing he says is that, we should have, if we're going to look at the effects of supplements, we need to do randomized controlled trials. We need to hold all drugs to the same standard. We need to do randomized controlled trials. So randomized controlled trials are like the poster child for pharmacological interventions, drug interventions. And what they are is you get a population of people, you split them into two groups, and you're going to have either your drug blablabanol or placebo. And you're going to give, whichever group one, drug blablabanol, and the other group the placebo. And you don't know who's getting what, they don't know who's getting what. And then you're going to look at some clinical endpoint like heart attack or myocardial infarction or something like that. And it's really a great design for that sort of thing, because, when you start off at baseline, everyone in the trial has same levels of drug blablabanol in their blood. Guess what? 0, because we don't make drugs. We don't eat drugs. It's not something that we're, eating on a daily basis. This is a pharmacological drug that's been designed with a certain mechanism of action to act on a certain thing, to do a certain thing, right? Like, statins, for example. So, you can do that where you have this randomized controlled trial for a certain X amount of time, and you look at the effect on heart attacks, and you don't have to measure anything. The problem is that you can't apply that same thinking for nutrition research, because everyone has different levels of vitamins and minerals in their body. People eat different diets. You have people that are, have inadequate levels of things, are deficient in certain things, have optimal levels of certain things. And so if you just take this random group of people, put them into two groups, and then give them a vitamin supplement, and then try to look at some clinical endpoint like heart attack, well, guess what? You're going to have people that were severely deficient, and whatever you gave them, they're still severely deficient. Or you're going to have people that have total adequate levels, and you're giving them a vitamin they already have adequate levels of, and you're trying to look at some clinical endpoint. You can't do nutrition research with the same mind frame, whereas we're going to do the same type of trial that we do for drugs. That's just, you can't do that. You can't do that. You have to think about the way you control and design a clinical trial. And when you're doing nutrition research, where you're looking at the effects of vitamins and minerals, first of all, there's two very different things going on. When you have a pharmacological drug, you're giving it to someone because they're already falling apart. Okay, they're already falling apart, and you're trying to help them not fall apart as fast by giving them something, that blocks X or Y or Z, like cholesterol synthesis for one, statins. With vitamins and minerals, these are things that are important for preventing you from falling apart. These are things that we require, these enzymes we need, cofactors for enzymes that we need to do like hundreds of different physiological functions, thousands of different physiological functions, So we're talking about a different start point, right? It's not like you give people vitamins when they're falling apart. No, they need to be getting their vitamins to make sure they don't fall apart. You know? And so the whole randomized control trial using so he kept saying this, like, we need to hold vitamins to the same standard as drugs. We need to have randomized control trials, and that's how we, determine whether or not they're effective or their efficacy, basically. But like I said, you can't do that. You have to measure people's vitamin and mineral levels. Let's say you're looking at vitamin D, at baseline to see how deficient they are. For one, this is going to determine the dose that you give them. If someone's like 12 nanograms per milliliter and you give them 400 IUs a day, they've even shown in that Annals of Internal Medicine paper that doesn't work. They're still going to be deficient at the end of the trial because, 400 IUs a day raises your blood levels by like 5 nanograms per milliliter. So that's just not the way to do nutrition research.
The difference being that giving someone something that's completely alien to the body but could be beneficial, like a drug, or giving them something that is absolutely essential to the body, that is necessary and is a part of normal everyday diet, that you can't look at the two of them the same way. That the drug may help people, but the reality is you're introducing something that's completely alien to the person's system in order to benefit them, whereas with vitamins, you are just regulating or measuring what is essential to the human body. And pretty much has been established that vitamin B, vitamin D, vitamin C, all these different things, various aspects of nutrition are essential to human health.
That's one aspect of it,
So you can't look at a drug the same way you look at vitamins.
You can't. And also, like I said, people already start off with varying levels of these vitamins. So when you're randomizing them into two different populations, you don't know. Some of these people could be very deficient. Some have plenty of vitamin, K or whatever you're looking at. So you need to measure them at baseline.
So you'd have to
get a deficient population.
Right.
You need to get a population that's low, because the whole point of giving someone a vitamin supplement is to bring them up to an adequate level. It's not to like have some effect on top. It's not like a drug where you're blocking some receptor and having some response. No, you're taking vitamins and mineral supplements because you're not getting everything you need from your food, and you want to get yourself up to an adequate level. And so what you need to do is start off. You need to measure people's levels at baseline. Start off with the population that's inadequate. Give them a vitamin and mineral supplement to get them to an adequate level, and then you can measure something. So it's really important, and I think I even talked about this last time, is quantifying these levels. Well, this is the reason why. You can't, starting off with the population of people that has enough vitamin D, and you're trying to look at the effects of vitamin D on cancer incidence, well, guess what? They already have enough vitamin D. They're already in this adequate range. Or conversely, if they're really, really, really deficient, and you're looking at the effects of vitamin D and cancer incidence, and you give them a dose that still makes them, they're still deficient, well, guess what? We don't know how vitamin D is affecting cancer incidence from that one study because they're still deficient at the end of the trial. So the conclusion is, my dose was inadequate to bring them to an adequate level of vitamin D. That should be the conclusion, not, vitamin D doesn't affect cancer incidence. Which is what people tend to do. They'll... you have to sell your story to get it published. Mm-hmm. You do years and years work of research, and you find this negative data. And it's like, well, you got to sell it somehow. So you're not going to sell it by saying, didn't give them an adequate dose, and they're still not, up to adequate level. So that's really the one first thing about Offit and so many others, including the people that wrote this whole "Enough is Enough" editorial. Yeah. They're the same. They were looking at randomized control trials. Most of the time, they started off with people, didn't measure anything at all at start or throughout, the follow-ups. And, it's like, well...
Well, the Enough is Enough was even worse, because the Enough is Enough, they were studying, two of the things they were studying were heart attacks in people over 65 and dementia delaying the onset of dementia in people who are over 65. Like, Jesus fucking Christ, you're talking about people who are dying.
Right.
And you're giving them vitamins and saying, didn't stop them from dying.
Right.
You're dealing with completely broken human beings that are like on death's door. They're forgetting shit. They don't know where they are, and their heart's failing. And you're like, vitamins don't work.
Yeah.
So to say vitamins don't work in those two circumstances is ridiculous. And their other circumstance showed a slight improvement with vitamins. And yet they still came to conclusion that vitamins don't work because that's the most salacious title.
Right. Vitamins and minerals are important to prevent disease. They are important to prevent disease. It's not like if you're already falling apart and you've already been deficient for years and years and years and years and years, guess what? It's much harder to patch things up, much more difficult. You've already acquired so much damage. You've acquired so many different things that are going wrong that trying to patch it up later is really difficult. And so, in those studies also, they did things where they were giving it to cancer patients. And like I said, or smokers. They were looking at the effects of beta-carotene or vitamin A in smokers. And the thing is that smokers are a different breed because, they're taking in poison every day. And what happens is their lungs, they have a very oxidative environment. So when they take beta-carotene, which is part of the vitamin A family, this beta-carotene gets cleaved because they have oxidative stress and stuff going on. It gets cleaved into some of these cleavage products, which actually damage DNA more and can accelerate cancer. But the thing is that that's specific to smokers. If you give the same dose of beta-carotene to a non-smoker, guess what? That doesn't happen. They don't get those cleavage products, and it doesn't start damage their DNA more, and it doesn't accelerate cancer. So, you have to really, the context is so important. Like, you can't just say, vitamin A is bad for you. No. Well, if you're a smoker, this is a certain context where, there can be problems with taking high doses of beta carotene for that reason, where you've got all these oxidative, stress things going on. And so they cleave the beta carotene to certain cleavage products that normal people don't get, so context is very important. And this is something that so many people so often ignore, including Offit. He's basically states that, and he says, it's clear and consistent that antioxidant supplements are bad for you and can give you cancer. And he gives two examples. The one example he says is the prostate cancer study. And the second example he says is because you need pro-oxidants around in your body to prevent cancer cells. It kills cancer cells, right? Okay, so it's kind of like, it's kind of a big overgeneralization, and it really shows me his lack of understanding of mechanism, either because it's too much work to look into it, because it's a lot of work, or, just because he doesn't care. So let's start off with the first part. He says, vitamin E can cause prostate cancer. This is huge. There was a huge study called the SELECT trial, where they took the selenium and vitamin E cancer prevention trial. Selenium is also important. It's a cofactor for about 25 or so different proteins that are, one of them being glutathione peroxidase and synthase and all these different antioxidant genes. And it's been shown to high selenium was correlated with low cancer incidence. So they thought that would play a role in prostate cancer. So they did this big trial where they got something like 30,000 men. Is pretty big. And they gave them supplements. They gave them either vitamin E. So vitamin E is actually, there's a whole family of vitamin E's. It's not just one vitamin. There's actually 8 different forms of it. And alpha, beta, gamma, delta. And the present is either tocopherols or tocotrienols, trienols. And the major forms are alpha and gamma tocopherol, okay? And these two different forms of vitamin E, the alpha and gamma tocopherol, they're antioxidants, but they actually have separate functions. So the alpha tocopherol is a very potent antioxidant. So it's very good at, getting reactive oxygen radicals that's generated by normal metabolism. These things damage your DNA, but they also damage your lipid, bilayer of your membranes, cell membranes. What happens is vitamin E is fat soluble, so it gets in those lipid bilayers, and it prevents that oxygen radical from damaging it. And what happens is if you don't have that happening, your lipid membranes get stiffer and stiffer over time. And this is part of aging, where they become rigid, and it's hard to transport metabolites into your cells. It's hard to transport proteins in and out. It screws up stuff. So it's important to have something like alpha tocopherol preventing that from happening. It also prevents your proteins from being oxidized, which causes problems. So that's really important to have, and you actually need to get it from your diet. The vitamin E's you have to get from your diet, from plants. We don't make it ourselves. The gamma form actually is also an antioxidant, but it's an anti-nitration one. So nitration is formed from your immune system. Just normal immune function, like, fighting off things, creates reactive nitrogen species. So these nitrogen species also do the same thing as oxygen species. They damage your DNA, they damage your lipid membranes, they damage proteins in your body. So having both alpha tocopherol and the gamma tocopherol, they're doing two independent functions, is important because they're making sure you're not getting those oxygen or nitrogen reactive species damaging crap in your body, basically. Okay? And that's important to prevent cancer, to prevent, a lot of, diseases of aging. So proteins that become oxidized or get nitrated aggregate, and they can form, things in plaques in your brain. They can cause neurodegenerative diseases. So it's important to have, these antioxidant mechanisms in play to prevent that from happening as we age. Okay? So with that said, the alpha tocopherol, and you can see how complex this is, the alpha tocopherol is the major form in your tissues and in your bloodstream. But the problem is that when you take really high levels of it, so the RDA is like 22.4 IUs. When you take really high, like 10 times that, like 400 IUs, what happens is it depletes your gamma levels. Okay? And this has been well known for like over a decade. Multiple labs have shown this, including the lab I work in. They showed it many years ago before I even joined their lab. So, taking high levels of alpha can be bad because it depletes the gamma, and the gamma has a separate function from the alpha. It prevents that nitration. It also is an anti-inflammatory, inhibits COX enzymes, which are involved in generating prostaglandins. So what they did was they gave men either alpha tocopherol, 400 IUs, so 10 times the RDA, maybe even more than 10 times, and selenium. Okay, so they gave them either alpha tocopherol, selenium only, or both alpha tocopherol and selenium, or the placebo. And then they followed up, they did a couple of follow-ups to look at prostate cancer incidence. Okay, so the first study was about a 5.5-year follow-up, and this was published in like 2009. And what it found was there's no effect. Taking vitamin E had no effect on cancer incidence. Taking selenium had no effect. But what you can see if you look at the data is they measured their alpha and gamma levels at baseline, and then at 5.5 years, what happened was those men taking 400 IUs depleted their gamma tocopherol by like 45% at the end of the follow-up, which is like crazy. That's not good. And then the second follow-up, what it found is it's 7.5 years later, they found that, oh my goodness, the men taking 400 IUs of alpha tocopherol had a 17% increased risk of prostate cancer. And then they went on to say, oh my goodness, 400 IUs of vitamin E a day can cause prostate cancer. However, the men taking the selenium with the vitamin E didn't get it. So the men taking that alpha tocopherol by itself, but took ones that took with the selenium with it, didn't get it. So the selenium protected, and they didn't know why. So another study then recently came out from this is all the same big cohort of people, found that only the men that were severely deficient in selenium to start the trial that took the alpha tocopherol were the ones that had the increased cancer incidence. So I started thinking about this. Well, why is that? Selenium also is important. Like I said, it's important for like 25 or so proteins. One of them is important for preventing nitration, reactive nitration products. So I was like, wow, well, this makes sense. You're taking high levels of alpha tocopherol, depleting your gamma levels, which is important to get rid of nitration damage, right? Which damages DNA, causes cancer, things like that. And yet you can give someone the selenium, or if they're not deficient in the selenium, that doesn't happen. And one of these selenium proteins also does that. So the bottom line is, does taking normal levels of alpha tocopherol, like 22 or 30 IUs a day, deplete your gamma levels? No. Is it good for you? Like, is it going to prevent your lipid membranes from oxidizing and your DNA from getting oxidized? Yes. Is it going to give you cancer? No. it's actually the opposite. It prevents cancer. Is taking high levels of alpha tocopherol dangerous? Well, it can be, yes, because it depletes your gamma. So, taking high levels of alpha tocopherol is not a good thing because it can deplete your gamma. But that's only if you're also selenium deficient. It's very, very complicated. It is not simple, and it's not as simple as taking vitamin E is going to give you cancer. No, there's a whole host of complex mechanisms that are at play here. And the reality is, if you want to, 60% of the population doesn't have enough alpha tocopherol, okay? 60%. So if you want to supplement with it, just don't be dumb. Take a lower level of it and supplement with the mix. You can buy mixed tocopherols together. So they get the alpha, the gamma, the beta.
What is a good source of that?
So avocados, pecans, walnuts, plant sources. I like to get my almonds also. So I get my vitamin E mostly from plant sources, and also I use almond milk, unsweetened almond milk in my smoothies. Like I said, I make a really big smoothie every morning, which has all these different micronutrients.
Unsweetened is the key, really.
Right.
Because I have a friend who said, "Oh, the almond milk's amazing. I'm not drinking milk anymore. It tastes so good." I go, "Okay. What is it? What kind of flavor is it?" And he goes, "Vanilla." I go, "Look at the fucking label, please."
Right.
I go, "What does it say as far as grams of sugar?" It
Right.
was 19 grams of sugar per serving.
Yeah, you have to get the unsweetened. But
Well,
it has a lot of magnesium in it and about a lot of the mixed vitamin E. So it's a good source.
So, vitamin E supplementation, in your opinion, you should probably just get it from diet to make sure that your levels are normal, healthy levels, and that it's the balance of the different types of vitamin E's?
Yeah, I prefer getting it from diet. There are mixed tocopherols that you can buy that are low. It's not like a 10 times the RDA, and they're mixed. So you're getting the gamma with the alpha.
Do you know of a good source for that?
I think there's Swanson brand may have a good one. Is that what you mean by a
like a
brand?
good company?
Yeah, and then that I think that might be a good one. I can't recall off the top of my head, but they are pretty reliable in general.
So in a sense, he oversimplified things, but there is a danger of taking too many vitamins when you're taking vitamins like a vitamin E that could potentially, if you're taking one form of it, that could potentially deplete your absorption of the other form.
Exactly. If you're taking a really, really high dose of the alpha tocopherol, vitamin E, there could be a potential danger in it depleting the gamma form.
And how would anyone know whether they're taking alpha or gamma if they just have a vitamin E?
If you have a vitamin E container and you look on the back of it, it'll say like alpha tocopherol. It'll say the sort, like it says alpha or gamma. Most supplemental forms are alpha, and that's because that's the predominant species found in tissues and in plasma. So people, we thought it was like the most important. But we found that gamma is also really important. But you can buy mixed tocopherols. You can buy mixed tocopherols.
Okay. So that is just one thing that he brought up that was incorrect.
Well, so that was one he said the data is clear and consistent. And for one, that's anything but clear. I mean, there's lots of complex things going on. And he said that it's clear and consistent that supplemental antioxidants cause cancer. Well, there's also other studies showing that, these people, in the case of prostate cancer, if you look at their blood levels of alpha, tocopherol, and gamma, those with the highest levels of alpha and gamma have the least, prostate cancer incidence. So there's inconsistencies in terms of what exactly is going on, and we're still really understanding trying to understand all the mechanisms at play. But he also made the overgeneralization that taking supplemental antioxidants is bad because you need pro-oxidants to kill cancer cells. And that was like the second part of his why supplemental antioxidants are bad. And, this is another example of context. So, if you don't have enough of the alpha, gamma, tocopherols, then you're going to have increased DNA damage. You're going to have, things that cause mutations. And what happens is you're going to, accumulate that over time, and that's going to lead to cancer. So not having enough of this vitamin E is not good. And like I said, 60% of the population doesn't have enough. But the flip side is that if you already have cancer, then taking supplemental vitamin E, what happens is because it prevents that oxidation, then there's mechanisms in your body that induce cell death. When you have oxidation, when you have this damage to your DNA, tumor suppressor genes get activated and they kill the cell. So what they've shown is that in mice, if you take mice that already, if you give them cancer and you give them supplemental vitamin E, you can attenuate that whole pathway that activates the death of these cancer cells. But that's not the case if you take a mouse that doesn't have cancer and you give them vitamin E. It actually prevents the DNA damage. So this is a case where you're looking at context. So if someone that doesn't have, doesn't have cancer in their body, you want to make sure they're not causing DNA damage, which is happening, every second, by making sure they have enough of these antioxidants. But on the flip side of that, if you already have cancer, taking a bunch of supplemental vitamin E is not a good thing because you can attenuate that pathway that is important to kill cancer cells. So it's another thing where it's like context is so important. You need to differentiate between people that are normal, healthy, and people that are trying to prevent themselves from getting cancer by preventing these things that cause cancer, which vitamin E can help prevent, versus talking about someone that already has cancer. And
so it's just incredibly irresponsible to make a simplified version of these incredibly complex processes that are going on.
In my opinion, it's a big overgeneralization, and it's probably because diving deep into this stuff it takes time and it's complex, and you need to understand how some of these things are interacting and working. So it's much easier to be like, oh, you read the study, the conclusions, and oh, yep, that's it.
That's the thing that people love, too. They love when someone can break it down in a clear sentence. Antioxidants can cause cancer. Well, let me get on Twitter and let everybody know that they're being an idiot.
Right.
I mean, people love doing that. They love saying things like that. Like nice, clear, concise, easy to digest. High alkaline diet kills cancer. Whoa, I didn't even know. I'm just going to fucking...
I don't think there was any possible way I could boil that down into like one sentence.
Didn't seem like you could.
Biology is very complex, and trying to oversimplify it doesn't necessarily mean you have a good understanding of the complexities.
This is certainly going to be a podcast that requires a notepad.
Yeah,
you're gonna have to do a lot of Googling.
Yeah,
remembering
the whole alpha tocopherol and gamma tocopherol. It's something I think is important for people to understand. It is alpha tocopherol is the major supplemental form, and the last study I saw said something about like 11% of the population takes high levels of alpha tocopherol. So you have a certain percent of the population that's going overboard, and they probably don't know about the effects that's going on. The depletion of their gamma and how that can be bad. So I think it is important for those people to realize that. But on the flip side, someone like Offit, his solution to that is he wants to have high-dose vitamins FDA regulated so that people like him who have an MD can prescribe them. And so that's his solution to, high-dose vitamins. And yeah, it's if you think about it, first of all, who's going to define what a high dose is? Is it the RDA? Because if you look at something like vitamin D, but right now the RDA for vitamin D is like 600 IUs a day, and 70% of the population doesn't have enough vitamin D. And taking 600 IUs a day, if you're deficient, isn't going to get you to an adequate level.
So that's not a good level. What is a good level of vitamin D? If 600 is not an accurate level, what's an accurate level?
So in a couple of studies that I've read, where people that were deficient in vitamin D took 4,000 IUs a day for a year, that was enough to get them up to 30 nanograms per milliliter.
It took a year?
It took a year.
Holy shit.
And
why does it take so long?
They measured it after a year. So it's possible if they had done some time points maybe in between that it could have raised their levels, higher than that.
Was it a slow absorption into the body or something? Like, why would it take so long to get the levels?
I'm not saying it's going to take a year. Mm-hmm. It's just the way the study was designed.
I see.
They measured it a year later. So it's possible that it didn't take a year, but the study was that they looked at it a year later after taking a year of 4,000 IUs a day. But that was enough. I think generally speaking, people that are supplementing between 2,000 and 4,000 IUs generally tend to have adequate levels if they're not severely deficient to start with. If you're severely deficient, it can take longer to get up to an adequate status.
What are the other things that Offit said that you didn't think were accurate or were problematic?
Yeah, so he also said that the data is clear and consistent that supplemental vitamins don't do anything. There's no positive benefit from them. That's what he said. No positive benefit. And like I said, to his credit, I haven't read his book, so maybe he goes into some specifics that I'm not aware of. However, to say that supplemental vitamins have no benefit is like, really? You know, I mean, they've even done randomized controlled trials showing that omega-3 fatty acid supplementation lowers all-cause mortality. So, you know, supplementing, and he actually refers to omega-3 fatty acids as an antioxidant, at least in that podcast he did. So, you know, that's one thing. Vitamin D supplements, people that have supplemented with vitamin D, 1,500 IUs a day. I don't remember how long they did that for, but they had it was many years. There was a 17% decrease in cancer risk, and there was a 30% decrease in cancer mortality, and like a 40% decrease in cancers of the digestive system, including colon cancer. So that's one for vitamin D. Let's see, what's another one? There was another study where they looked at women that took multivitamin. And but this was not a controlled trial. This was they did a questionnaire and found out like how frequently women took multivitamins and how many times a day or how many days a week they took them. And what they found is that the ones that took vitamins on a daily basis had the longest telomeres. They measured telomere length. So that's another one. Magnesium. So we're talking about, you know, taking supplemental vitamins helps you fill some of those nutritional gaps that you're not getting from your diet. And the reality is that we're not getting a lot of those vitamins and minerals from our diet. We talked about this a little bit last time.
Is it possible to eat a completely healthy diet and get everything you need, or do you need to supplement? I mean, is it possible?
That's a good question. I don't know. I mean,
like things like vitamin D. What would you have to do? I mean, I know it's in milk. For vitamin D, you'd have to really be living in a place that was exposed to the UVB during winter and summer, and you'd have to be out in the sun for 15 minutes, you know, because getting it from fish, well, maybe fish has a good bit of it, but you'd have to eat fish every day. And also to get the omega-3s, you'd have to eat fish like every day. The omega-3 fatty acids are another big one. And you'd have to eat quite a bit of fish, right? And certain fish like salmon.
Like fatty fish, right, exactly. You'd have to eat like fatty fish. So I think it would be really difficult. Like I personally, I agree with Offit and that goofball Dunning in some instances where, you know, you should try to get all your micronutrients much as you can from your diet. I agree, you know, and, you know, case in point, I do my smoothie. I definitely try to eat my greens and healthy meats and fish. I really like fish. But I do supplement. You know, I take omega-3 fatty acids. I take vitamin D. I take a multivitamin, which has my, you know, selenium and some of my trace elements, my iodine. And what else do I take? I also take a B complex, even though most people aren't. They don't have low levels of the B vitamins due to like fortification and stuff now. I actually take a B complex because it's really kind of interesting. But our lab has shown that. It's partly shown that as we age, I talked about how your lipid membranes get more rigid, and this includes your mitochondrial membranes. They get more stiff, and over time, what happens is metabolites and stuff can't get transported as easy. And also, what happens is these proteins, they bind cofactors. Like B vitamins are really important for a lot of proteins in the mitochondria that are necessary for metabolism to generate ATP. And these proteins require B vitamins, and a lot of them are embedded in the membrane. So when the membrane gets stiff, the protein binding constant changes to that B vitamin. And it's been shown that you can overcome that, meaning you actually can overcome that, messing up that binding constant because it screws up the structure of the protein by increasing the level of B vitamins.
So what level do you recommend? Like what's the USD RDA, and what do you think should it be?
For B vitamins, you know, there's no telling what it should be. There's just no empirical data to show how much more you would need to overcome that.
How much do you take?
I have a B complex, and I just can't recall off the top of my head because there's so many of the B vitamins.
Do you know what the name of your B complex is that you take?
Right now,
company?
Right now, I have one from Vitacost, but I've been switching over to Swanson brand because I
Spell that Swanson like
SWANSON. They're pretty reliable. They've been around for a really long time. They generally have some reliable supplements. For omega-3, I like Nordic Naturals. They're really good.
And Nordic Naturals, what that's a fish base.
Fish oil.
So there's some oils. Here's the Swanson. It has 25 milligrams of riboflavin, which is 1,471% of the U.S. RDA. Thiamine, which is B1, it has 25 milligrams, which is 1,667% of the U.S. RDA. Vitamin C, it has it as well. In the B complex, it has vitamin C, which is interesting.
Yeah, that's interesting.
833% of the U.S. RDA. And the other big one, vitamin B6 is 1,250%, and vitamin B12, 2,083% of U.S. RDA. That's interesting. I've always found that interesting when you look at some vitamins, you look at multivitamins, and you know what the RDA is, and you see their percentages fucking off the charts.
Yeah.
They have 2,000% of the U.S. RDA of B12.
Yeah.
They're
that's
just saying
a lot.
the U.S. RDA is a knucklehead. I mean, that's what they're saying in their company. They're
Well,
saying, look, fuck them.
In the case of the B vitamins, like I said, most people aren't deficient in them, and they haven't really shown any toxicity in taking too much of these B vitamins, and they are water soluble, so you end up peeing them all out. I personally am taking them just because I've seen some of this data where it shows that some of these proteins that are when your your lipid bilayer is kind of more rigid and gets screwed up, over time, and also when your oxidation does that, or eating rancid fat, things like that. Changes some of the structure of those membrane proteins that require B vitamins. And like I said, I don't know how much more you need. It'd be cool to do a study to figure that out.
But it's not a dangerous one because they're water soluble.
It's not, I haven't seen any data that shows it's a dangerous one, for one, because they are water soluble and you pee them out.
What are the fat soluble ones that we should be careful about overdosing?
The alpha tocopherol is one.
Which is E.
Right. Like I said, the whole interplay between the gamma and alpha, is a...
So gamma tocopherol, alpha tocopherol, all the E's, those are fat soluble.
Right. E.
Okay.
Also,
so those you should be careful.
Vitamin D, you can, you can take too much. If you take... The toxicity levels that I've seen have been shown at like 10,000 IUs a day for some time. That anything over that, and actually I think it even is quite higher than that, but that's the limit of toxicity.
What about omega-3s and omega-3s in plant-based form versus in fish form?
Great question. So if you look at the omega-3, there's EPA, there's DHA, and ALA. So the major plant form, like flaxseed oil or flaxseed, walnuts, for example, have ALA. Okay, and they don't have EPA or DHA. Now you can convert a very small percentage of ALA into the EPA, but it's like 5% of it. It's very inefficient conversion, and women may be able to do a little bit better of a conversion. I can't recall off the top of my head what their number was, but the point is that if your only source of omega-3 is flax or walnuts, you're not getting your DHA and EPA.
You hear that, vegans?
Yeah, that's the vegans. If you're a vegan, really, I just microalgae oil. I mean, the phytoplankton are what making these omega-3s. The fish eat the phytoplankton; it gets concentrated in fat. Microalgae oil, I recommend for people that are vegetarian, vegan.
Microalgae oil, but when you're eating microalgae oil, what totally constitutes vegan? Is there any sort of animal protein in microalgae oil?
I guess they're phytoplankton. So if you're vegan based off of philosophy, where you don't want to eat like any type of creature,
phytoplankton is kind of a creature.
It is. It's kind of a creature, but. So that I guess that could be a problem. But if you're a vegetarian or vegan for health purposes, and there's not like a philosophical component where you have a problem with eating these phytoplankton, then
vegans shouldn't they avoid? There's certain probiotics that are. That's a creature too.
True. bacteria.
Probiotics
Yeah.
is living life forms.
Yeah. No, that's a good point. I don't know.
They move. I mean, they're active.
They are active. They are absolutely active.
Just because you can't see them, I mean, you're basically eating little tiny turtles or something. You know, I mean, whatever.
So
I mean, the little moving thing.
Right. You know, I don't know what the philosophy, because I don't have a lot of vegan friends
Good
to
for
really
you.
know.
Cut those people out of your life. It's very important.
I don't have anything against them. I just, you know.
I don't either, but I like fucking with them.
So, but the the DHA and EPA are really important. They're really,
Yes.
really important. And so you asked, can you take too much? I mean, for one, you know, EPA is it's got a very potent anti-inflammatory effect, which is, you know, important for a variety of different mechanisms. You know, inflammation, chronic inflammation over time can lead to a lot of age-related diseases. And DHA, as we, I think, even discussed last time, that's a really important component of your cell membranes, particularly in the brain, like 40% of it. So,
I take a lot of fish
I take a
oil.
lot. I take a lot too.
How much do you take a day?
So, well, it depends. So you have to look, okay, you got your fish oil container, and on the front it says like 1,000 milligrams, or if mine says 2,120 or something. And then you turn it around the back and you look at the breakdown. Okay, how much EPA, how much DHA, and then there's other fish oils mixed. So on my container, it says like 2,100 and something. And I turn it on the back, it's got 800 milligrams of EPA and 600 milligrams of DHA. So, and then all the rest is just fish oil stuff. So I actually calculate how much I take based on those components, the EPA and DHA, because that's important. That's what's important to me. So I take about 6 pills of those a day. And a serving size, I guess a serving size is 2. So
yeah. I take a little bit more than that. I take 10.
10 pills?
Yeah.
Yeah, okay. And that's pretty cool. I've done that. I mean, I've done that in the past.
It has a big impact on joint health.
Yeah.
It has a big impact for grapplers, for jiu-jitsu people. We're always getting sore knees and sore elbows. It's a big issue. And even sore backs. And it has a huge impact on that.
No.
The anti-inflammatory
Absolutely.
reaction.
Absolutely.
What is the anti-inflammatory? What is the actual mechanism, the anti-inflammatory mechanism involved in fish oil?
So the EPA is the major part. So you can actually buy EPA, like just, you know, there's companies that'll sell it. If you're really just looking at the anti-inflammatory part of the fish oil, you can buy EPA. It's a little more expensive. But what it does is it inhibits that whole arachidonic acid pathway, which produces prostaglandins. So it's like upstream where it's inhibiting the production of arachidonic acid, and then prostaglandins, which activate COX, and like all, you know, you got this whole cascade of inflammation going on. It plays a major role in that. There's probably other mechanisms that I haven't even read about, you know, other things going on as well that I'm not even sure, you know, feedback mechanisms, stuff like that. But that's the major way that EPA does. And they've even shown that like taking 2 grams of EPA a day can reduce your C-reactive protein levels. Oh gosh, I don't remember the exact amount by how much it was, but it was pretty significant, where it's like it really, you can measure it, lowers inflammation, systemic inflammation.
How many thousand milligrams are in a gram?
You just said it.
1.
1,000 milligrams are in 1 gram.
So 2,000 milligrams is 2 grams.
Yeah.
Why don't they just call it a gram? Why do they have to call it
I don't know.
a milligram?
Yeah. I usually
Once it
call
gets
it.
to 1,000.
Yeah. It's like, it's much easier. Yeah. It's a
Why are
gram.
you confusing me?
Maybe because people aren't aware of the whole metric system and stuff. So
Yeah.
if they see 2 grams, they're like, oh, well, that's nothing. That's
Right.
just 2.
Well, people get confused because we go ounces all the time with so many different things.
That is confusing.
Why? I mean, I don't understand why we use the metric system in some cases. In some cases, we don't at all.
Right.
Like liters. Oh, it's a liter. Well, how many quarts is in there? Oh, fuck. I don't know.
Yeah, because
like, why is there a quart and a liter?
Right.
What are you doing to me here?
well, in the lab we do everything milliliter, liter. I mean, so
Gallons when you go to the gas station.
I don't even. Yeah, I don't even that. I always have to Google how many.
Inches. Well, try fucking. I'm 90 centimeters. What the fuck is 90 How tall is that? I don't even know. You know what I mean? If someone like tried to guess someone's height in centimeters.
Yeah.
Like,
So is your fish oil that you take, is it both EPA and DHA?
That's a good question. I take this Carlson's fish
I've
oil.
taken that
Yeah.
before. Do you
Yeah.
take the lemon flavored one in the bottle, or do you take them in pills?
I take both. I bring the pills with me when I go on the road. I take the lemon in the bottle at home.
Ah, okay. Yeah, there is something to be aware of with the omega-3 fatty acids, and that's because they're polyunsaturated fatty acids. They're very prone to oxidation. So keeping them at 4 degree, keeping them in the fridge, sorry,
Yeah.
lowers that oxidation process, and also just smelling it, take a sniff of it and make sure it's not rancid smelling when you're taking it, because that's the one thing with the omega-3 fatty acids. I mean, like I said, I take a lot of omega-3 fatty acids, and I think they're really important. But I think it's important to be aware that they can go rancid, and if they go rancid, consuming them can be not as good.
Yeah. And when you leave them in the refrigerator, how long are they good for when you crack open that bottle? I've always given it a 2-week window. If I don't drink it in 2 weeks, I throw it out. Is that...
I don't use the bottle anymore, but the bottle is more open to be to oxygen. So that is one thing to be aware of with the bottle. And
You
that
just
was
go with the pills?
I do. That was something that I think that was part of the reason why I switched to it. That, and I think I got really sick of that lemon
Yeah.
flavor.
It gets kind of funky.
It gets kind of much. Yeah. So
It's easier to just not taste anything and you swallow the pills, but then you're dealing with the gelatin as well.
Right,
But
but
gelatin's
gelatin-
not good. Bad
I
for
haven't
you.
convinced myself that it's doing my charm, but who knows? Maybe I haven't dug deep enough.
What about krill oil? How do you feel about krill oil?
I haven't really done much reading on the krill. Is that has vitamin A as well, or that's cod? I'm thinking I'm conflating
Cod oil. Cod liver oil was the one that they always gave us when we were kids, and everybody was like, "Bleh, get that away
I
from
don't know
me."
if I've ever tried it. Like the taste. I think I've had a pill and I couldn't really
You know
tell.
what? I actually think I have krill oil here. Hold on a second. Let me pull this out.
Yeah, but taking the omega-3 and just popping it open every once in a while and smelling it is a good idea. I recommend doing that because you can smell what rancid fat smells like. So that's what I like to do. Every once in a while, I'll just grab my omega-3 pill out of the fridge and take some scissors, cut it open, and smell it. And if it doesn't smell rancid, it's still good.
Yeah, I've got some krill oil here. Tell me about that. Well, krill oil, I don't know what's supposed to be the benefit of krill oil over fish oil.
It doesn't have a lot of DHA or EPA in it.
No?
I mean, it says 75 milligrams of DHA and 130 milligrams of EPA, and that's 2 soft gel servings.
So you think that fish oil is probably a better choice than krill oil? Well,
I don't know if there's also 1 gram of krill oil. So it says 1 gram of krill oil. Maybe there's something in there I'm not aware of because I just haven't done a lot of research on it. But I'm looking at the DHA and EPA specifically. If you're taking this to get DHA and EPA, I would say this is not a very high amount.
So you think that if there is something good about krill oil, you should probably... Why is there these things? Krill oil radically better than fish oil? Blood sugar regulator?
I don't know anything about it, unfortunately.
Maybe there's some other reasons.
Maybe there's something else. Yeah, like I said, I'm completely ignorant on that.
It says here on this website, krill oil actually influences your metabolism and genes to improve. The reference study found that although both fish oil and krill oil contain omega-3s, they differ greatly in how they affect the genes controlling your metabolism. Krill oil enhances glucose metabolism in your liver, whereas fish oil does not. Krill oil promotes lipid metabolism, whereas fish oil does not. Krill oil helps regulate the mitochondrial respiratory chain, whereas fish oil does not.
So DHA does do those things. So when they're saying fish oil...
So what are they talking? They're talking shit.
Well, I'm not sure. I'm not sure I'm following.
I'm not either.
So because DHA does regulate lipid metabolism, and also DHA and EPA, in addition to the anti-inflammatory effects of EPA, and in addition to the lipid membrane part of the DHA, these fatty acid molecules are signaling molecules that actually bind to different DNA regions in your gene and activate them, much like vitamin D does. And DHA and EPA do this. So DHA can activate genes. So, if they're talking about, and it's been shown to activate mitochondrial metabolism genes. So when they say krill oil
does
can,
and fish oil does not. They're just
it doesn't make
incorrect.
any sense to me.
Okay.
You have to be specific. What is it are you talking about DHA? Because they're in both, what else is there?
Yeah, this is an article from Mercola. Is that a good website?
Mercola is hit or miss. I think he does have a lot of good. Every once in a while, there's something, it's both. I haven't spent a lot of time reading his stuff, but I do think he does have some good information. But sometimes
so it doesn't sound like krill oil's bad, but it does seem like fish oil is probably more beneficial or provide. Well, there's other things in here
what's
too.
in it. Yeah. So the point is, you have to be specific. If you're saying just krill oil, well, you're talking about other things in the krill oil, because if we're talking about gene activation, DPA and DHA is doing that. DHA is activating genes. That's what it's doing, in addition to what it's doing for your lipid membranes.
Yeah, this is so confusing after reading what you said, because it's saying some studies have shown that krill oil may be 48 times more potent than fish oil. It means you'll need far less of it than fish oil. It's confirmed by a 2011 study. 48 times more potent in what sense, though?
Yeah. What are we talking about in the krill oil? I just don't understand.
I don't either. That's the problem. And people have mortgages. They have jobs. They have children. They have dogs to feed. They have a plant to water. They don't have fucking time to get into all this stuff.
You have to... I feel bad for people. I mean, it's hard enough for me.
But you're a person. What are you talking about?
I mean, no, it's because it's craziness. It's craziness. And the reality is understanding mechanism. Like when I'm saying mechanism, I'm talking about DHA can activate, promoters in certain genes to increase the expression of mitochondrial metabolism genes. That's been shown. And so when you say krill oil is better, can do that and fish oil can't, that makes no sense to me. You need to tell me what specifically is in the krill oil that's not in fish oil, because, DHA and EPA are in fish oil.
Right.
Are we talking about a concentration dependent thing? I mean, is there more DHA and EPA in krill oil? I don't know, actually.
Yeah,
So.
I don't. I think this is something that's going to have to be really studied in length. And I would love to see you dive into all these claims. But there's quite a few claims online about the benefits of krill oil over fish oil. But it doesn't. One of the things is saying krill oil contains acetaxan. Ooh, boy, try this one. ASTAXANTHIN. Astaxanthin.
Say it again.
ASTAXANTHIN.
Yeah, that's a mouthful.
Astaxanthin.
I'm not familiar with it.
A unique marine-sourced flavonoid that creates a special bond with the EPA and DHA, which allows direct metabolism of the antioxidants, making them more bioavailable. Does that make sense?
No.
Does that sound like malarkey?
Possibly. It doesn't make
Possible
sense.
malarkey.
Yeah.
Yeah. God, that it really is the issue, isn't it? There's just so much to try to sift through, find out how to separate the wheat from the chaff, as it were.
It's, with the DHA and EPA, the really important thing, in my opinion, is getting enough of it. Like taking a good bit of DHA and EPA, because most people aren't getting any of it. And all this other stuff, I'm just not sure how significant it is. Mm-hmm. Maybe it does increase bioavailability of a little bit. I don't know if it really makes a big difference.
So for the folks that are taking they're getting their omega-3s just completely from a plant source, what can they do, if anything, to try to?
I really think the microalgae oil is the best.
That's
I mean, I would recommend taking the fish oil, but I guess they won't do that. Some cases, vegetarians won't do that. So the microalgae oil is the next best thing, in my opinion. And like I said, I don't really know much about the krill oil, but I do know that these DHA is very important, as is EPA. So for a variety of different reasons.
Well, I'm going to send you some of these studies on the krill oil to see if unless you get bored reading it and you can't do it. It seems like there's just such a it's such a mess. There's so many different supplements, and just going into all the contradictory arguments, the back and forth about all these different ones, it can be incredibly taxing. Your website is a great resource of this. if people want to go to foundmyfitness.com and try to figure out what you've already sort of described and you've already explained, but how does a person start?
I think a really good resource that I like is the Linus Pauling Institute. So if you go to the Linus Pauling Institute, they are pretty good about writing it's a scientific research institute that's associated with Oregon Health University or Oregon State Health University, something like that. But they do a very balanced review on a variety of different supplemental vitamins and minerals and essential fatty acids, and that they give you both sides. And now, if they go into the krill, sometimes they'll go into things like that. But generally speaking, I really like the Linus Pauling Institute. It's a good resource.
Are there places where someone can go where they can, say you're not sure what you need? Like we were talking about, like various levels of vitamins and getting them tested. Is there like a standard place where you could go where you could, like someone who lives in the middle of nowhere can find a place and get their blood tested?
I think one of the companies that I am familiar with is Wellness FX. And I have no association with them other than I did a couple of guest blog posts for them where I, for free, wrote about vitamin D and magnesium. And they actually, I think they're pretty much in almost every state now, where you can go onto their website and they have a variety of different assays they'll do. Well, they'll measure different vitamins and minerals and omega-3 fatty acids. They'll measure different things, C-reactive protein. Mm-hmm. And you can go to any... enter your address and go to like a lab corp around nearest by or whatever and get your blood drawn. And then they'll give you the data within a couple of days. They'll help you interpret it. They have a variety of different physicians and nutritional people that can help you interpret what your blood results mean. So like I used it myself. We also got a test for my mother-in-law to use, and she lives in Mississippi. So, and it's pretty much, I think it's pretty much everywhere now. So I really like them. And obviously you can go to your physician, your primary healthcare provider, but they may not measure everything that you want, like omega-3.
And they also might give you some of that, all you need to do is eat a balanced diet.
Right.
How many times have I heard that from a physician who looked like shit?
Overweight physician. I mean,
sloppy, loose skin all around his face.
Right.
Like, dude, you're melting.
You're telling me what to do?
Yeah.
Yeah. So, but the Wellness FX is pretty cool. I really like them. But the nutrition thing, I think it's making its way into medicine, and that's part of the reason why people like Offit, who is an MD, these people, they've been trained very differently. They haven't been trained in understanding preventative medicine, understanding how these complex micronutrients are interacting with different, proteins in our body and how that's important to prevent different diseases. They're not thinking about it from the same frame of mind, as people like me that are PhD researchers or nutritionists, and there's a variety of other, naturopathic doctors maybe have more. There's a whole, variety of different.
Naturopathic gets lumped into homeopathic sometimes accidentally.
They're different, yeah.
Yeah, completely different. I've been guilty of it myself.
Yeah, I think I have as well.
I confuse them in a way where anything homeopathic I think of as horseshit.
Isn't homeopathic like that crazy thing where they like dilute things to like crazy amounts where you can't even, there's like no active compound. If you Google it or look at the Wikipedia
of what is the official term?
Homeopathic. They have their own measuring system too, I think. It's like
really?
They'll take a compound and they'll dilute it like a millionfold. And it's like to the point where there's like no biological activity and they give it to people. I'm pretty sure that's homeopathy. I didn't realize that until recently.
Yeah, repeatedly diluting a chosen substance in alcohol or distilled water, followed by forceful striking on an elastic body. What? Dilution usually continues well past the point where no molecules of the original substance remain.
Is that so? Okay, let me tell the story. So I recently had my wisdom teeth removed, and my oral surgeon gave me this case of pills that was arnica. It's like
nonsense.
Well, I thought, no, it was homeopathic, which really makes it nonsense. There is some antimicrobial activity and things like that in arnica, the real arnica plant.
But the homeopathic arnica is like sugar.
It's water.
Yeah.
It's water, literally. Like, but I didn't know it was homeopathic at first. I just thought, oh yeah, it's just arnica. And it wasn't until like my husband looked at it and he's like, do you realize this is homeopathic? I was like, what's that? I didn't really even, I knew homeopathic was kind of crazy. And we looked it up and it was like mind blowing. I couldn't believe that they were giving, I don't even think he knows. I think he probably made the same mistake I did where I just thought it was just arnica.
This is for anybody defending homeopathy. Homeopathy, this is on Wikipedia, this is all with references, lacks biological plausibility, and the axioms of homeopathy have been refuted for some time. The postulated mechanisms of action of homeopathic remedies are both scientifically implausible and not physically possible. Although some
There you go.
clinical trials produce positive results, systematic reviews reveal that this is because of chance, flawed research methods, and reporting bias, which is pretty common.
So there you have it, homeopathy, really.
Yeah, I mean, anytime someone says homeopathy, they usually are wearing crystals, and they know someone who's a channeler.
Right. So, no, getting back to my wisdom teeth, though, it's a kind of a cool topic. I had to get them removed because they were like impacted and causing problems and pain and such. So I did some research. I was like, God, if I have to get my teeth removed, there got to be some kind of benefit from it. And I found that our wisdom teeth have something called dental pulp stem cells in them. And these dental pulp stem cells, so this stem cell research is like a whole, I'm really excited about the stem cell research field and where we're going with that. But anyways, our wisdom teeth have dental pulp stem cells in them that can actually form other tissues in our body, like bone, cartilage, and they even showed recently they've taken dental pulp stem cells from people with impacted wisdom teeth, taken them out and transplanted them into mice that had damaged motor neurons, and it was able to differentiate into damaged neurons, neural type cells, and help replace that. So I went and looked online to see if anyone was banking them because they do cord blood banking, where you can bank your cord blood. And indeed, there was a company that, there's a couple of companies that are both associated with cord blood banks as well, but they bank the dental pulp stem cells so that you can, when you have your teeth removed, they send you a kit with like buffered saline solution. Your oral surgeon will stick the teeth in them, and then they ship it off back to the company and they preserve it in liquid nitrogen. They kind of do very minimal processing. They don't actually remove the stem cells. They keep it in the tissue, the dental pulp tissue, and they freeze it in liquid nitrogen so that you can later use it if you need it. So it's actually really cool because you can use this if you have damaged cartilage, bone, possibly Parkinson's disease where you need to replace damaged motor neurons. So that I thought that was pretty cool. It's like a benefit because getting your wisdom teeth removed is not fun at all.
Yeah, that is fascinating, though.
Primary teeth from children also. So in the children's teeth, they're losing their primary teeth, and you just throw the teeth away. Mm-hmm. They have a really rich source of dental pulp stem cells.
Wow.
So you can, your child loses their tooth, and you can bank it, where you freeze it. It's like $625 for the whole processing, and then to store it, it's like $125 a year. I personally think it's a great investment if you have kids and they are losing their teeth, or also if you have your wisdom teeth and you have to get them removed.
Yeah,
why not?
My daughter's about to lose her first tooth, so that's perfect.
Yeah, the company that I went with, I can give it to you. I actually talked to their cell biologist. I was on the phone because I read through all the procedures in these scientific papers to see exactly how you process it in the optimal way and the best way to have the best viability after you thaw them. And so I was like getting on the phone. I'm like, okay, do you do this? Do you do this? And so I went with this company because I spoke with one of their cell biologists, and they had done the things that I thought were the best. So anyways, the whole stem cell field is cool. Yeah, it's what's really cool is that they can take fibroblast cells from your skin, like skin cells that we slough off like every day, and they can add 4 different transcription factors, like 4 different genes that they can add by like a viral, and add virus to them, and they can reprogram them to become these pluripotent stem cells in your body, meaning they can form any type of cell in your body. So you can take a skin cell and you can make it, into a brain cell, a liver cell, a heart cell. I mean, this has huge, huge implications for regenerative medicine, but I think also just for extending lifespan.
What did you think about this latest study where they took mice? And did you read about this, where they took the blood of young mice and they reintroduced it to old mice?
Yeah. So that Was that a recent study, or was that? I think,
It was the recent findings.
because I've seen other studies that were not super recent, but what they did was they took like bone marrow cells. They did a bone marrow transplant and took, which obviously bone marrow forms your blood cells, from young mice and transplanted them to old mice, and the mice like lived longer.
Yeah, this is recent. It's it's from March 6th or May 6th rather. They actually took an injection of young blood. So they took the blood of they took the blood of young mice. They injected it into old mice, and they had tremendous benefits, including regenerating different cells, brain cells, tissue cells.
Oh, really? Brain cells as well?
Yeah.
I mean, it makes sense that you'd regenerate different blood populations because you have, stem cells, stem cells in the blood and stuff that are.
It improved the performance of elderly mice in memory and learning tasks.
Wow, that's pretty
fucking
cool.
Crazy. Structural, molecular.
Will you send me that paper?
Yeah, absolutely.
Yeah, that's awesome.
Yeah.
I mean, this is the stuff I'm really excited about now. Is this the stem cell research and reprogramming? Where the epigenetics is a really cool part where you can reprogram your cells to basically be younger. They're finding now that so epigenetics. I think we talked about this a bit last time. Refers to changes in gene expression, and things like methyl groups and acetylation groups will sit on top of your DNA and turn genes on and off. But now what they're finding is they're trying to look at patterns of methylation, like in your DNA. So they've already solved the Human Genome Project, where they know genes, and now they're trying to look at the methylome, the human methylome, and they've been able to, over the past few years, they've been able to identify that there's patterns of methylation in your genes that happen with age. And they've even been able to systematically identify. They've taken blood from people, various ages, like from 19 to 101, and they've been able to identify the age of the person, the chronological age of the person, based on their methylation patterns, like with 96% accuracy. And they've been able to do this within 4 years. I think they'll be 4 years off, plus or minus 4 years. So they can take someone's blood cells, look at their methylation pattern, and say, you're 50 years old, and the person will be, between 50 and 54, or 46 and 50. So I think that's pretty freaking amazing.
It is amazing.
And the thing that's really cool, I'm on this kick, this epigenetics kick, where they've been able to now also look at the cancer cells, like in a person. They'll take a tumor sample, and then a tissue from the same person, another non-tumor tissue, and they'll look at the methylation pattern, and they'll see that the cancer or the tumor tissue ages by 40% based on the methylation pattern.
Wow.
And yeah, so it's like the cancer cells aging rapidly. So what's really interesting is now they're looking at what genes these methylation patterns are happening around, and they're finding it's like DNA repair, mitochondrial metabolism, antioxidant genes, like everything that we've been talking about in this whole podcast, things that affect DNA damage and metabolism, all these things. Methylation patterns are happening clustered around these genes. And the cool thing about it to me is that if we're figuring this out, then we can figure out how to reprogram our cells to be young and extend lifespan. And I really think that we're getting close to doing this. So, I mean, if you think about, I'll give you an example, like stem cells. Stem cells also have methylation patterns that are very distinct to stem cells. And there's certain genes that are, when a gene is methylated, it's not being expressed. There are certain genes that are not expressed in stem cells for a reason, because when they get expressed, they cause the cell to stop dividing. And you don't want your stem cell to stop dividing, because stem cells are what's repopulating the tissue, okay? And they found that a certain gene is methylated in young people. So when we're young, our stem cells have, this gene's methylated. But as we get older, the methylation goes away. So there's enzymes that actually are called demethylases that take off the methyl groups. And this gene becomes active, and then the stem cell stops dividing. It's like you lose. You're basically losing your stem cell. And then, more stem cells you lose, the worse off you are. You can't replenish your damaged tissue and all that. But what they found was that the thing that activates that thing that takes off the methyl group is something called NF-kappa B, which is an inflammatory. That thing is activated by inflammation. NF-kappa B, inflammation activates NF-kappa B, and then it activates this whole pathway of demethylases that take off methyl groups. So what I'm thinking is that inflammation is a chronic signal. It's a way that I've been able to link environment and the way, when I say environment, I just mean like damage, constant accumulation of tissue damage, the DNA damage I was talking about, environment to epigenetics. So it's like you have a chronic signal of inflammation. It's activating these demethylases. They're taking methyl groups off of DNA, and now you're expressing genes that are usually not expressed when we're young in stem cells that stops the stem cell from dividing. So it's like a really cool link between, environment and epigenetics that, as it relates to aging, which we know that environment regulates epigenetics. So to me, it's kind of cool because as we're figuring out these programs, what that means is that I think that we'll be able to reprogram our cells to become young.
That's incredible. How long do you think it's going to be before that happens?
I think that we're getting really close with. I mean, if you look at, like I said, with the stem cell research that we're doing now, where we can even take renal epithelial cells that we excrete in our urine and make it into a pluripotent stem cell. We can make it become a cell in our liver. So I think with the advances we're making with that, in combination with this figuring out the human epigenome, where we're looking at methylation patterns and figuring out what's happening with age, I think we're going to make huge strides in the next decade. I mean, hopefully, if funding doesn't go down, it's been going down the drain. But
has it been?
Yeah. Oh, yeah. I mean, funding is hard. It's really hard getting funding in science because.
But why is it with something like anti-aging? It would seem like, boy, if there's something to be made where there's money to be made, it's funding anti-aging research. Seems like, God, that's the way to go.
Well, there is the National Institute of Aging. Funds a lot of the aging research, and there's also some private foundations that are funding it. But the reality is that we're funding it with taxpayer dollars, right? I mean, there's not a lot of money for research in general. Period. I mean, you'd think that cancer one would be another, right? I mean, the NCI is funding a lot of cancer research, but the problem is that it's hard to get funding. And the things that are funded, here's what I see as the big problem. The things that are funded by these big institutes are typically things that are already been proven. They're not as creative. They're things that people feel are already solid. So it's like, oh, you already published on this. Yes, we're going to fund you on that. As opposed to someone that has this very creative idea. It's very risky. It's like, well, I'm not going to, there's all these other people that are trying to compete for this money. Giving funding for something like that is risky. So they don't fund a lot of creative research, which unfortunately is what makes leaps in science.
This mice thing is so fascinating because they showed that after 4 weeks, stem cells in both the areas of the muscle and the brain got a boost of activity and were better able to produce neurons and muscle tissue. And then they also discovered that injecting the old mice, or rather the young mice, with old blood was a huge setback. When conjoined to an older mouse. So bringing the old blood into the new mouse, the creation of new cells in the young mouse slowed, and old blood seemed to cause premature aging.
Yeah,
isn't that incredible?
It is incredible. I mean, it's.
But how long before people start fucking doing something gross, like taking a person, stealing their blood, introducing it to their own to stay young? I mean, it's really vampirism.
Yeah, I mean.
For sure.
Oh, wait, no, I remember that.
Someone has read this.
I think I remember that study now. I'm going to grab another water real quick.
Yeah, please. This one right there. Well, this study, it was published on in the Washington Post. The article was published on May 4th. And the study, two of the studies published online, the Journal of Science, came out... let's see here. Yeah, one of them was from Stanford, the Stanford group. Well, they apparently they've been working on this for quite a while, but the results of the blood results have been pretty shocking to a lot of people.
I think a lot of it comes down to, like I said, there's these. If you look at the epigenome, the methylation patterns in these stem cells, these young stem cells, even young cells in general, they're very different from old ones. Yeah. And that affects the way, I mean, if you're looking at epigenetics, you're talking about regulating a whole host of genes, like hundreds of different genes. And so if these things are being differentially regulated when you're young versus old, then, taking someone's young blood and transplanting into the old, transplantee would make sense, because now you're basically taking all those patterns that we've been able to identify, and gene expression things are now going back to young. So it's like now you're not expressing genes that are causing your stem cells to senesce. You're expressing more things that are involved in DNA repair and things like that.
Yeah.
So
it's just incredible what they were able to do. They actually had more endurance. They navigated mazes faster, ran longer on treadmills, and they easily outperformed their control peers.
That's awesome.
Who were only given saline.
Yeah.
So it's like a definitive response.
Right.
But
And
it's such a creepy one.
I think this is something that we can do with the reprogramming of our own like skin cells into like pluripotent stem cells.
Right. And that's really fascinating. And
it's the same concept, but you don't have to get someone's young blood. You're doing it to yourself.
And haven't they been able to come up with artificial blood cells? Isn't that another thing that they've been working on?
That sounds familiar. I don't know. Artificial heart cells as well, I think. Yeah. I don't remember exactly the studies and the mechanisms.
It's just so much. Well, although it is difficult to get funding, there are so many different. Yeah, artificial blood. Wow, patient ready. This is on from the-scientist.com, and it's saying that wow, this is incredible. In the midst of news that engineered organs are being implanted into animals and people, researchers announced the creation of artificial blood for transplant. This is very recent too, April 16th. It's nuts. We live in strange times when it comes to these things. Like every day or so, it seems like some new study from somewhere in the world is popping up that shows this incredible breakthrough. The young mice study, this study, the artificial blood study. If they can engineer some sort of a super potent blood and introduce it into your body, I mean, it's similar to like what the cyclists did when they were blood doping. They would take their own blood, pull it out, inject it back into their body so they had more blood, or EPO, which stimulated
red blood cell production.
Yeah. That kind of stuff is just it's so trippy. The idea of sort of hacking and retweaking the components of the human body.
I agree, but I do think it's really cool. I mean, I know when I first got into the biological sciences, so I was a chemistry major in college. I did research using these nematode worms, C. elegans worms, that have like a 14 or 15-day lifespan. And they have a lot of the same genes that we have, but I could like inactivate one of their genes and literally double their lifespan. So they went from living like 14 or 15 days to like 30 days. And so it was you inactivate insulin growth signaling, insulin growth factor. And what happens is this growth factor then usually when it's active, it keeps this FOXO gene, which is a transcription factor, outside of the nucleus and doesn't allow it to perform all of the functions it usually performs, which is involved in a bunch of stress resistance, like hundreds and hundreds of genes. And so when you get rid of that IGF-1 signaling in the worms, FOXO gets activated and all those genes involved in stress resistance get activated, and the worms live twice as long. Wow. So it's like literally a genetic program in these worms that's controlling the way they age. So when I remember that hit me, it was kind of like, holy crap. Like, that's pretty cool. Like, being able to reprogram the way you age.
It's just such an amazing time with all this stuff because it seems like we're around at just the right time to catch this just incredible percolating of all these new studies and all these new things that are being developed. It's just such a strange time to like try to pay attention to all of it and watch it all happen. It's so exciting. It must be really exciting for you because this is like your field of study.
Oh no, it's great. I mean, I think we're definitely going to live longer.
How long do you think you're going to live?
Oh, in my lifetime, I don't know.
Without
reprogramming. Oh, without you mean just like
Yellowstone erupting or asteroid impacts.
I don't know. I'd like to be a centenarian.
That's it. 100? Good with 100?
Yeah,
But what if 100 comes around and they've got shit locked down where you look like you look now at 100?
And then I could go for 200.
Well, I feel like I'm happier now than I was when I was younger. I'm smarter. I understand stress better. I understand all sorts of emotions.
Right.
I understand management of my
Right.
body, of my hormones, management of my feelings, management of my energy levels, stress, relaxation techniques, stretching, yoga, all that different stuff. I feel like I'm just way happier. And you said that old expression, youth is wasted on the young. You know, like I understand life better. I'm better at it now. I'm better at being me. It was like it was an awkward thing when I was younger. I'm way more relaxed and easy with it now. I feel like at 200, I'd be a goddamn wizard. I would have shit down to a science.
Right.
I would have the amount of errors that I'd make as far as, everyday average things that you fuck up. You would fuck up almost nothing. Every day would be like you could learn new things. You could challenge yourself. You could constantly learn languages. Like I never understand the people that say like, "Oh, I get bored. I wouldn't even want to live past a certain age." Like, I don't know, but I never get bored. There's always new things
Yeah.
to learn, new things to
agree.
study, the new things to. The world is so big. There's so much to learn.
It would be awesome to just have time to sit around and learn how to play the piano, learn how to speak a new language. And I'm always sitting around reading about science, which I really enjoy doing. But I agree. I mean, there's so much out there to learn. And,
You
how
might get
can
to a
you?
point if you're 200 years old, you might abandon science totally and just fucking go down some totally different road. You might be a sculptor. You might, you know what I mean? I mean, you could live several lives.
I
if you're
certainly
200
hope
years
that I'd
old.
still be active. When I lived in San Diego, I was a surfer and surf instructor, and I used to
Were you
surf.
really? You
Yeah.
instructed people?
I used to teach at an all-girls surf school in La Jolla
wow,
Shores.
I love La Jolla.
Yeah,
Such
I
a
used to
great
live
town.
2 blocks from the beach, so I'd walk to the beach
It's
before
on
class.
fire right now. Do you see all that stuff about San Diego?
What? No,
You haven't seen?
my family's there.
Oh my God, San Diego's experiencing crazy fires right now.
No, I had no
Yeah,
idea.
Huge tornadoes of fire, 100 feet high.
Well, I haven't
Photographs.
gotten a phone call, so that's a good idea. I mean, good sign.
Well, I mean, the fire departments down there are on top of it because they have to deal with this stuff every couple of years. You know, there was the big one a couple of years ago. I was scheduled to go down and do a show, and there was these big fires, and it was so crazy. I wound up doing the show, but I said I donated all the money to the Red Cross. I'm like, I can't. I mean, there was so many people lost their houses. I just felt like it would be so gross
Yeah.
to go down there and just make money.
Yeah, I remember back in I think it was 2002 or something when we had a really big fire in San Diego, and I was actually out surfing. I was in the water, and it was really weird because there were all these pelicans and all these birds flocking to the water. I'm sitting out on my surfboard looking out on the horizon waiting for the wave, and it's like the apocalypse. Like birds were just coming to the water, right? And it's like, what is going on? So I get out of the water and there's just ash falling, because my car I was in La Jolla Shores at the time, my car was in the parking lot. And there was just ash all over the cars and parking lot. But it was that moment where I was sitting on my board and all the birds came to the water. I had this almost oh my God, it's a nuclear warfare or something that, that eerie moment. Have you ever had one of those where
Yeah.
you just get this eerie feeling where it's it's the end of this is it, And anyways, that fire burned a lot of really close to where my dad lives, actually.
I've been evacuated twice out here.
Really?
Yeah. My
Yeah, I guess
neighborhood came really close 2 times to giant fires. I mean, we have fires that are so bad out here. One time, while we were doing it, and this was the time I got evacuated, I was filming Fear Factor, and I drove from Los Angeles to Tejon Ranch. Tejon Ranch is about an hour and a half outside of L.A., and as I was driving near Simi Valley, which is about 40 minutes from here, Simi Valley is where the fire kicked in. And then from there, for the next 50 minutes of driving, there was no fire. Well, when I got to work, we started filming, we were doing the show, and then when we were done and we left, the fire had reached us. So the fire had reached, an hour of driving. So we're talking about, somewhere around 60 plus miles. And the entire right side of the road was like a Lord of the Rings movie. It was like I was expecting a demon to ride a flaming
That's
horse over
terrifying.
It was insane. For an hour of driving, all you saw to the right side of you was flames. It was amazing. The only thing that separated it was the highway, and flames were jumping the highway, and they had tried to keep the fire as far away from the roads and far away from structures, but it was so out of control that it traveled 50 miles plus in a few hours.
I mean, in a place like, this, like LA or San Diego, where there's a lot of dry, brush and like what kind of mechanisms do they have to, I mean, they obviously know this can be a serious problem. Is there any sort of like...
There's not much they can do. I mean, they try to stop it. They create fire breaks. They drop repellents on it. They drop this red stuff that squashes the fire like in certain areas, but this fire leaps. The problem is these things cross highways. The embers fly through the air. They land on dry brush and poof, they're off to the races. We're running out of time. You wanted to promote something. What do you got going on? What's your iPhone app?
I do. I have a new iPhone app. It's Found My Fitness, and it was just released like earlier this week. And it's basically you can get my podcast where I talk about my information, and also I have a news section where I give new news stories, and it's a community section. If you ever used Reddit or Hacker News, it's just based on that. So it's like that. Yeah, I'm trying to make an interactive community. So if you download my podcast, it would be really cool. Right now it's new, so it counts as twice the download.
Beautiful.
And also foundmyfitness.com is where you can follow me. I've got a bunch of gadgets on there you can click to download my newsletter, follow me on Twitter. I also have a Patreon campaign where I'm trying to do these podcasts, and I have a couple of milestones where I'm trying to do one, two podcasts a month or 4. So I'm asking people to donate, or not. It's not really donate, to pay, to pledge 25 cents a month to help me reach my milestones, so that I can help give you the context that you need and the mechanisms for how science and health and nutrition, all these things are interacting.
That's about as reasonable as you could ever ask. 25 cents.
Yeah, I'd like to have a lot of people just doing 25 cents a month, because it's it's less than a cup of coffee.
What? 1 minute? 1 minute to go. All right, we gotta wrap this up. So Found My Fitness at Twitter, Found My Fitness, foundmyfitness.com. All the information is there. Thank you, Rhonda Patrick. As always, you're amazing. These conversations are so enlightening and so educational. I mean, I got to go back over it 30 or 40 times to really absorb it all. But really, really appreciate it. Let's do it again in a few months.
Awesome. Let's do it.
Thank you so much. All right. Thank you to everybody tuned in. Thanks to our sponsor. Thanks to Stamps.com. Use the code word JRE and save yourself some money. Lots and lots of cool guests next week. I'll tell you about them all later. I'll see you soon. Steve Maxwell's coming back next week. Alex Ross from sharkworks.com. We're going to have a lot of fun. See you soon. Bye.